Lecture 2. The concept of the hallmarks of cancer provides a framework for understanding the complex biological processes that transform normal cells into malignant ones. This framework was first introduced by Douglas Hanahan and Robert Weinberg in 2000, with subsequent updates recognizing the evolving understanding of cancer biology. Cancer cells exhibit several key characteristics that allow them to grow uncontrollably and spread, leading to the disease's hallmark behaviors. These hallmarks can be divided into original hallmarks, emerging hallmarks, and enabling characteristics, all of which are interconnected in the development and progression of cancer. Starting with the original hallmarks, cancer cells thrive by sustaining proliferative signaling. Normally, cell growth and division are tightly regulated by growth factors that bind to receptors on the cell surface, triggering internal signals that promote cell division. Cancer cells either produce these growth factors themselves or modify their receptors to be constantly active, leading to continuous cell proliferation even in the absence of external growth signals. Evading growth suppressors is another hallmark where cancer cells bypass the body's natural checkpoints that halt cell division. Tumor suppressor genes like 53 normally act as brakes on cell division when DNA damage is detected. But in cancer, these genes are often mutated or their function is inhibited, allowing cells to divide unchecked. Resisting cell death, or apoptosis, is crucial for cancer cells. Apoptosis is programmed cell death that removes damaged or unnecessary cells. Cancer cells develop mechanisms to avoid this fate, often through mutations in genes like BCL2, which regulate apoptosis, thus enabling the survival of cells that should otherwise die. Enabling replicative immortality is essential for cancer's endless growth. Normal cells have a limited number of divisions due to telomere shortening, but cancer cells reactivate telomerase, an enzyme that rebuilds telomeres, allowing cells to divide indefinitely. Inducing angiogenesis is how tumors secure their own blood supply. Without sufficient nutrients and oxygen, tumors cannot grow beyond a certain size. Cancer cells secrete growth factors, like VEGF, that stimulate the growth of new blood vessels directly towards the tumor. Activating invasion and metastasis allows cancer to spread beyond its original site. Cancer cells alter their adhesive properties and become motile, allowing them to break away from the primary tumor, invade surrounding tissues, enter the bloodstream or lymphatic system, and establish secondary tumors elsewhere in the body. Moving to the emerging hallmarks, reprogramming of energy metabolism reflects cancer's shift towards glycolysis, even in the presence of oxygen, a phenomenon known as the Warburg effect. This metabolic shift supports rapid cell proliferation by providing the necessary building blocks for new cells, even if it's less efficient in terms of energy production. Evading immune destruction involves cancer cells developing strategies to hide from or suppress the immune system. Tumors can express proteins that inhibit immune cells, or they might trick the immune system into thinking they are normal, thus avoiding an immune attack. Finally, the enabling characteristics pave the way for these hallmarks to develop. Genome instability means cancer cells have an increased rate of mutations, which can lead to the activation of oncogenes or the deactivation of tumor suppressors, driving the hallmarks. Inflammation within the tumor microenvironment can promote cancer progression by providing growth factors, survival signals, and even fostering angiogenesis. The tumor microenvironment itself is a dynamic ecosystem where interactions between cancer cells, stromal cells, immune cells, and the extracellular matrix can either suppress or enhance tumor growth based on the balance of these interactions. These hallmarks are not isolated. They interact in complex ways. For instance, genome instability might lead to mutations that promote angiogenesis or alter metabolism. Inflammation can both result from and contribute to immune evasion. The tumor microenvironment influences how cancer cells signal grow, evade, death, and metastasize. This interconnectedness means that therapies often target not just one hallmark, but attempt to disrupt several pathways simultaneously, acknowledging cancer's multifaceted nature. Understanding these hallmarks not only deepens our comprehension of cancer biology, but also guides the development of more effective targeted treatments, aiming at the root causes of cancer's ability to grow and spread. Metabolic reprogramming in cancer represents a pivotal shift, where tumor cells alter their metabolism to support their aggressive growth survival under harsh conditions, and evasion of the body's defenses. This concept is central to understanding how cancer cells differ from normal cells in their energy production and the utilization of nutrients. Why metabolic reprogramming occurs. The primary reason cancer cells reprogram their metabolism is to generate both the energy and the biosynthetic intermediates necessary for rapid cell division. This includes the production of nucleotides, amino acids, and lipids, which are essential for new cell formation. Under conditions where nutrients and oxygen are limited, such as in the core of a solid tumor, this reprogramming allows cancer cells to survive and proliferate where normal cells would perish. Additionally, maintaining redox homeostasis is crucial for cancer cells. They generate high levels of reactive oxygen species, ROS, due to their rapid metabolism, which could be detrimental if not managed. By altering metabolic pathways, cancer cells can detoxify ROS, reducing oxidative stress and preventing cell death. How metabolic reprogramming occurs. Mutations in metabolic enzymes. Mutations in genes encoding enzymes like succinate dehydrogenase, SDH, fumarate hydratase, and isocitrate dehydrogenases 1 and 2, idhone IDH2, can lead to significant metabolic changes. For instance, mutations in idhone IDH produce oncometabolites that not only alter cellular metabolism, but also epigenetic regulation, affecting gene expression patterns that support tumorigenesis. Influence of oncogenes and tumor suppressors. Oncogenes like MyRA and PI3K can drive metabolic changes by increasing nutrient uptake and shifting metabolism towards glycolysis. The Warburg effect, even in the presence of oxygen. Meanwhile, tumor suppressors like 53 can regulate metabolic pathways to prevent cancer. Mutations in these genes remove this control, further promoting metabolic reprogramming. Cancer as a genetic disease. Cancer fundamentally involves genetic alterations. Mutations, amplifications, or deletions in the genome can lead directly to metabolic reprogramming by altering the function of metabolic enzymes or pathways. These genetic changes contribute to evasion of immune response. By altering their metabolic profile, cancer cells can become less visible to the immune system or even use metabolic byproducts to suppress immune cells, aiding in immune evasion. Interaction with the microenvironment. The metabolic changes in cancer cells influence and are influenced by the tumor microenvironment. For example, by consuming high amounts of glucose, cancer cells can starve surrounding normal cells, creating a niche where they can thrive. The acidic environment created by lactate production from glycolysis can also disrupt normal tissue function and immune response. The interplay between genetic alterations and metabolic reprogramming underscores cancer's complexity. It's not just a matter of uncontrolled growth, but involves a sophisticated rewiring of cellular functions to support survival, proliferation, and spread under conditions that would normally be hostile to cellular life. This understanding has led to the development of therapies targeting these metabolic vulnerabilities, aiming to disrupt cancer's ability to sustain itself through these altered pathways. In the intricate landscape of cancer biology, beyond the original and emerging hallmarks, there are additional layers that contribute to the complexity of the disease. These include non-mutational epigenetic reprogramming, unlocking phenotypic plasticity, the role of microbiota, and the phenomenon of cellular senescence, all of which play significant roles in cancer development and progression. Non-mutational epigenetic reprogramming refers to changes in gene expression that do not involve alterations to the DNA sequence itself. These changes can include modifications to the DNA packaging, histone modifications, DNA methylation, patterns and alterations in non-coding RNA levels, all of which can influence how genes involved in cancer are turned on or off. This reprogramming allows cancer cells to adapt their behavior without the need for new genetic mutations, providing a rapid means of evading treatments or environmental pressures. For example, epigenetic changes can silence tumor suppressor genes or activate oncogenes, fostering an environment conducive to cancer growth. Unlocking phenotypic plasticity is another critical aspect, where cancer cells gain the ability to switch between different cellular states or identities. This adaptability can be seen in cancer stem cells, which can transform into various cell types within the tumor, contributing to tumor heterogeneity and resistance to therapy. Phenotypic plasticity involves not just genetic or epigenetic changes, but also dynamic interactions with the tumor microenvironment, allowing cancer cells to respond to threats like chemotherapy or immune attack by altering their characteristics. The influence of microbiota on cancer introduces an external biological factor into the equation. The human microbiome, particularly in barrier tissues like the gut, skin, and respiratory tract, can profoundly impact health and disease. The microbiota can modulate the immune system, influence inflammation, and even directly affect cancer cells through metabolite production or microbial translocation. For instance, certain bacteria can promote inflammation, which in turn can drive cancer progression, or they might metabolize drugs, altering their efficacy in cancer treatment. On the flip side, a balanced microbiome might offer protective effects against cancer by enhancing immune surveillance or reducing chronic inflammation. Cellular senescence, traditionally viewed as a protective anti-cancer mechanism, presents a dual role in cancer biology. Senescence can act as a barrier to cancer by stopping cells that have undergone DNA damage or telomere shortening from proliferating further. However, the senescent cells do not just sit idly. They secrete a variety of factors known as the senescence-associated secretory phenotype, SASP. This secretion can influence the surrounding microenvironment, potentially promoting cancer in nearby cells through inflammation, angiogenesis, or altering the extracellular matrix. Thus, while senescence can suppress tumor initiation, the persistent presence of senescent cells within a tumor can paradoxically contribute to its progression, or even lead to the emergence of new tumors. These additional hallmarks and characteristics are intricately connected. Epigenetic reprogramming can lead to phenotypic plasticity, allowing cells to adapt in ways that might be advantageous for survival or metastasis. The microbiome can influence these epigenetic landscapes indirectly through chronic inflammation or directly through microbial products that affect gene expression. Senescence, in turn, can alter the local environment in a way that either supports or opposes cancer growth, depending on the context. Understanding these layers adds depth to our comprehension of cancer, not just as a genetic disease, but as a multifaceted disorder influenced by a symphony of biological signals within and around the cells. This interconnectedness underscores the need for multifaceted therapeutic approaches, targeting not just the cancer cells, but also the environment they create and exploit. Mitochondria, often heralded as the powerhouse of the cell, play a pivotal role in cancer biology, particularly in how they influence metabolic pathways like the tricarboxylic acid, TCA, cycle, and oxidative phosphorylation, OXPHOS. These organelles are not mere bystanders but active participants in the metabolic reprogramming seen in cancer cells, affecting both energy production and biosynthetic pathways essential for tumor growth and survival. The TCA cycle, also known as the Krebs cycle, is central to cellular metabolism, where it oxidizes acetyl-CoO, derived from carbohydrates, fats, and proteins, producing NADH and PHA-2. These electron carriers shuttle electrons to the electron transport chain in the mitochondria, which drives OXPHOS, the process where ATP is synthesized from ADP using the energy from these electrons. In cancer, this coordination between the TCA cycle and OXPHOS is often altered. While the Warburg effect describes cancer cells' preference for glycolysis, even in the presence of oxygen, not all cancer cells abandon mitochondrial function. Instead, mitochondria in cancer cells can adapt, supporting both high glycolytic rates and maintaining aspects of oxidative metabolism, a phenomenon sometimes referred to as hybrid metabolism. This metabolic versatility has made mitochondria a focal point of cancer research. From 2000 to 2022, over 30,000 scientific papers have explored mitochondria's role in cancer metabolism, highlighting their involvement in energy production, redox balance cell signaling, and even apoptosis regulation. Mitochondria can become dysfunctional due to mutations in mitochondrial DNA or nuclear genes encoding mitochondrial proteins, which can contribute to cancer by disrupting normal metabolic regulation, increasing reactive oxygen species, leading to genomic instability, or altering the cellular response to stress. The clinical translation of this extensive research has begun to reveal potential applications in cancer treatment and diagnostics. One significant area is the development of new therapeutic targets, drugs that target mitochondrial processes like inhibitors of mitochondrial complex I or modulators of the TCA cycle are being investigated for their ability to selectively kill cancer cells by either inducing metabolic catastrophe or resensitizing cancer cells to apoptosis. For instance, targeting enzymes such as IDHONE2, which are frequently mutated in cancers like glioma, could disrupt the cancer cell's metabolic advantage. Moreover, mitochondria offer a unique opportunity for non-invasive imaging in cancer detection and monitoring. Techniques like positron emission tomography, PET, with tracers that target mitochondrial activity, can reveal metabolic hotspots indicative of tumor presence or activity. This approach not only aids in diagnosing cancer, but also in monitoring treatment responses by tracking changes in mitochondrial function, providing insights into whether a therapy is effectively altering cancer metabolism. The connections between these aspects are profound. Mitochondrial metabolism influences the tumor microenvironment, affecting immune responses, angiogenesis, and even epigenetic changes through metabolite signaling. The dual role of mitochondria in promoting cancer growth, while also being a potential Achilles heel for cancer cells, underscores the importance of understanding these organelles' behavior in cancer. As research continues to unravel these complexities, the potential for mitochondria-targeted therapies and diagnostics expands, offering hope for more personalized and effective cancer treatments. This integration of mitochondrial research into clinical practice showcases how basic science can inform and transform medical approaches, potentially leading to breakthroughs in how we understand, detect, and treat cancer. Lecture 3. The history of cancer research is a long, winding journey through time, marked by pivotal discoveries, shifting paradigms, and an evolving understanding of the disease. From ancient observations where cancer was described in Egyptian papyri around 1600 BCE to the modern era, each step has contributed to our current knowledge. Early on, cancer was largely considered an untreatable fate, but with the Renaissance came the first anatomical understandings leading to surgical interventions. The 19th and 20th centuries saw the advent of microscopy, revealing cancer at the cellular level, followed by the identification of carcinogens, the discovery of oncogenes, and the development of chemotherapy, radiotherapy, and later targeted therapy. This historical timeline underscores a shift from viewing cancer as merely a local growth to recognizing it as a systemic disease influenced by genetics, environment, and metabolism. This brings us to a contemporary debate encapsulated in the concept of cancer as a metabolic disease, a perspective that challenges the predominant genetic model of cancer. The idea that cancer might fundamentally be a metabolic disease posits that the primary driving force behind cancer is not just genetic mutations, but rather a profound alteration in cellular metabolism that supports uncontrolled growth and survival. This concept was first notably advocated by Otto Warburg in the 1920s, when he observed that cancer cells preferentially utilize glycolysis for energy production, even when oxygen is abundant, a phenomenon known as the Warburg effect. His work suggested that this metabolic shift could be both a cause and consequence of cancer, rather than an incidental byproduct of genetic changes. This metabolic perspective has gained traction with research showing how cancer cells adapt their metabolic pathways to meet the high demands of proliferation, survival under stress, and metastasis. They reprogram their metabolism to ensure a constant supply of energy and biosynthetic precursors, often by increasing glucose uptake, altering the TCA cycle, and shifting towards aerobic glycolysis. This metabolic reprogramming isn't just about energy. It also influences the tumor microenvironment, immune evasion, and resistance to therapy, creating a feedback loop that further supports cancer progression. The connection between this metabolic view and the historical timeline of cancer research is evident in how each era's understanding has built upon the last. Initially, cancer was seen through the lens of surgery and local treatment. Then, with genetics, the focus shifted to mutations in oncogenes and tumor suppressors. Now, by integrating metabolic insights, we recognize that these genetic changes can lead to or result from metabolic dysfunction, suggesting a more holistic view where metabolism is not just an outcome, but a driver of cancer. This paradigm shift has profound implications for both research and clinical practice. It opens up new avenues for therapy, focusing not just on killing cancer cells, but on disrupting their metabolic advantages, perhaps through diet, drugs targeting metabolic pathways, or even exploiting the metabolic differences between cancer and normal cells for diagnostic and therapeutic purposes. It also rekindles debates on prevention where lifestyle and environmental factors influencing metabolism might play a significant role in reducing cancer risk. The journey from ancient observations to today's complex understanding of cancer as potentially both a genetic and metabolic disease reflects science's iterative nature. Each era's discoveries lay the groundwork for the next, showing that in cancer research, as in the disease itself, the whole is greater than the sum of its parts, with metabolism emerging as a crucial piece of this intricate puzzle. Otto Warburg was a pivotal figure in the field of biochemistry, whose work has left an indelible mark on cancer research. His most notable contribution came in the form of the Warburg effect, which he described in the 1920s. Warburg's research initially focused on cellular respiration and the role of mitochondria, for which he was awarded the Nobel Prize in Physiology or Medicine in 1931. However, his observations on cancer cells led him to propose that these cells exhibit a unique metabolic behavior even when oxygen is plentiful. The Warburg effect describes how cancer cells prefer to use glycolysis to generate ATP, converting glucose into lactate, rather than completing the oxidation of glucose through the TCA cycle, and oxidative phosphorylation in the mitochondria, as normal cells do. This metabolic shift towards aerobic glycolysis, fermenting glucose to lactate under aerobic conditions, was groundbreaking because it suggested that cancer might not just be a genetic disease, but also a metabolic one. Warburg hypothesized that this metabolic reprogramming was not merely a consequence of cancer, but could be a cause, suggesting that cancer arises from defects in cellular respiration. The details of the Warburg effect reveal much about cancer cell behavior. By favoring glycolysis, cancer cells secure several advantages, they can rapidly generate ATP for their high-energy needs, produce biosynthetic intermediates necessary for rapid proliferation, and survive in hypoxic environments, often found in the core of solid tumors. This metabolic adaptation also contributes to the acidification of the tumor microenvironment due to lactate production, which can break down surrounding tissues, promote angiogenesis, and help cancer cells evade the immune system. The implications for cancer behavior are vast. The Warburg effect not only supports cancer growth and survival, but also plays a role in resistance to therapy. Cancer cells that rely heavily on glycolysis might be less vulnerable to treatments that target oxidative phosphorylation, thus providing an escape route for cancer cells from conventional therapies. In clinical practice, the Warburg effect has been harnessed for diagnostic purposes, notably through positron emission tomography, PET imaging, the use of ATNF fluorodeoxyglucose, ATNF-FD, a radioactively labeled glucose analog, leverages the high glucose uptake characteristic of cancer cells due to their glycolytic metabolism. ATNF-TG. PET scans can highlight areas of increased metabolic activity, thereby identifying tumors or metastases based on their avidity for glucose. This method not only aids in cancer detection, but is also crucial for staging, assessing treatment response, and monitoring for recurrence. The connection between Warburg's early work and modern clinical applications, like PET imaging, illustrates how foundational scientific discoveries can translate into practical medical tools. The Warburg effect has not only shaped our understanding of cancer metabolism, but has also opened avenues for both research into new therapeutic strategies, targeting metabolic pathways, and for improving cancer diagnostics and monitoring. Warburg's legacy, therefore, lies not just in his Nobel-winning mitochondrial research, but in how his observations on cancer metabolism continue to influence oncology, offering a lens through which we view cancer's complexity and potential vulnerabilities. The metabolic landscape of cancer cells diverges significantly from that of normal cells, primarily characterized by a shift in energy production and biosynthetic capabilities. This divergence centers around the utilization of oxidative phosphorylation, OXPHOS, versus glycolysis. In normal cells under aerobic conditions, glucose is completely oxidized through the TCA cycle in mitochondria, leading to OXPHOS, which is highly efficient in ATP production. However, cancer cells often exhibit a preference for glycolysis even in the presence of ample oxygen, a phenomenon known as aerobic glycolysis, or the Warburg effect. Aerobic glycolysis in cancer cells specifically involves converting glucose into pyruvate, which is then rapidly reduced to lactate rather than entering the mitochondria for further oxidation. This metabolic switch might seem counterintuitive since glycolysis yields significantly less ATP per glucose molecule compared to OXPHOS. However, this less efficient pathway offers several strategic advantages to cancer cells. Firstly, the high glucose uptake compensates for the lower ATP yield. Cancer cells can consume glucose at a much higher rate than normal cells, ensuring they have enough ATP to support their accelerated proliferation. This increased glucose consumption can be detected and utilized in clinical settings, like in PET scans, where cancer cells' avidity for glucose highlights their presence. Secondly, the glycolytic flux helps avoid pyruvate accumulation. If pyruvate were to build up due to high glycolytic rates, it would potentially inhibit glycolysis itself. By converting pyruvate to lactate, cancer cells maintain the glycolytic process, so momentum ensuring a continuous supply of ATP and intermediate. Moreover, the accumulation of biosynthetic intermediates is perhaps one of the most critical advantages. Glycolysis not only produces energy, but also generates precursors for biosynthesis. These intermediates, like glucose 6-phosphate fructose, 6-phosphate and 3-phosphoglycerate, are diverted into pathways that synthesize nucleotides, amino acids and lipids, all essential for the rapid division and growth of cancer cells. This metabolic reprogramming supports the biosynthetic demands of cancer cells, allowing them to proliferate at rates far exceeding those of normal cells. This metabolic adaptation also influences the tumor microenvironment. The production of lactate acidifies the surroundings, which can disrupt normal tissue function, promote angiogenesis, and create an immune-suppressive environment, aiding in immune evasion. Furthermore, this metabolic shift can contribute to resistance against therapies that target mitochondrial function or rely on oxidative stress to kill cancer cells. The differences in metabolism between normal and cancer cells thus not only define how cancer cells grow and survive, but also present both challenges and opportunities in cancer treatment. Understanding these metabolic pathways has led to the exploration of new therapeutic strategies, such as targeting glycolytic enzymes or exploiting these metabolic differences for selective drug delivery or imaging. The Warburg effect, once seen as a metabolic inefficiency, has revealed itself as a sophisticated survival strategy that provides cancer cells with the resources they need to thrive under conditions where normal cells would fail. In the intricate dance of cellular metabolism, certain enzymes play starring roles, particularly in cancer cells where metabolism is reprogrammed to support rapid growth and survival. One such key enzyme is pyruvate kinase, PK, which catalyzes the final step of glycolysis, converting phosphoenolpyruvate, PEP, into pyruvate while simultaneously generating ATP from ADPT. This reaction is pivotal because it not only produces energy, but also determines the fate of pyruvate, either entering the mitochondria for oxidative phosphorylation or being converted to lactate, as is often the case in cancer cells following the Warburg effect. In healthy cells, pyruvate kinase exists in multiple isoforms, with PK-MLMAIN being the predominant form in tissues like muscle and heart, where it supports efficient ATP production through the oxidative pathway. However, in cancer cells, there's a switch to the PKM2 isoform. PKME2 exhibits unique regulatory properties. It can exist in a less active tetrameric form or more active dimeric form, influenced by various cellular signals. This isoform's ability to be modulated allows cancer cells to fine-tune their metabolism. When PKM2 is in its less active state, it can lead to an accumulation of glycolytic intermediates upstream, which are then shunted into biosynthetic pathways, supporting the high demand for nucleotides, amino acids, and lipids needed for cell division. Visualizing these metabolic pathways through schematic diagrams helps illuminate the contrasts between healthy and cancerous metabolism. In a diagram depicting glucose metabolism, you'd see glucose entering both types of cells, but the paths diverge significantly. In healthy cells, glucose would be processed through glycolysis, with pyruvate entering the mitochondria for the TCA cycle and OXPHOS, producing a high yield of ATP. The diagram would show PKMON as the gatekeeper of this process. Contrastingly, in cancer cells, the diagram would highlight the dominance of PKM2, which might be depicted in a less active state, leading to a bottleneck at the end of glycolysis. Here, the pyruvate would be shown converting to lactate in the cytosol, with arrows indicating the redirection of glycolytic intermediates into biosynthetic pathways. This visualization would emphasize how PKM2's regulation allows cancer cells to conserve intermediates for growth rather than solely for energy production, showcasing the metabolic flexibility and advantage cancer cells gain. These diagrams would not only show the biochemical reactions, but also the implications for cellular behavior. For instance, the increased lactate production would be linked to the acidification of the tumor microenvironment, affecting immune response and tissue invasion. The diagrams would ideally connect the dots from basic biochemical reactions to broader cancer biology, illustrating how a single enzyme like pyruvate kinase can influence cancer's aggressive phenotype. Understanding these key enzymes and reactions, particularly through visual aids, underscores the complexity of cancer metabolism. It reveals how seemingly subtle changes at the enzymatic level can lead to profound physiological effects, providing insights into potential therapeutic targets. By targeting PKM2 or manipulating its activity, one might disrupt the metabolic advantage cancer cells, enjoy potentially slowing growth, or sensitizing them to other treatments. This connection between enzyme regulation, metabolic pathway alterations, and cancer progression encapsulates the essence of cancer as not just a genetic, but a profoundly metabolic disease. Lecture 4. Pyruvate kinase is an enzyme at the heart of cellular metabolism, catalyzing one of the last steps in glycolysis, where it facilitates the transfer of a phosphate group from phosphoenolpyruvate (PEP) to adenosine diphosphate (ADP), yielding adenosine triphosphate (ATP) and pyruvate. This reaction is crucial because it not only generates energy in the form of ATP, but also dictates the metabolic fate of pyruvate, determining whether it will be further oxidized in the mitochondria or converted to lactate. In healthy cells, this process is regulated to optimize energy production, but in cancer cells, the dynamics shift significantly. Schematic diagrams of glucose metabolism can vividly illustrate these differences. In such a visual aid, one would see glucose entering cells, proceeding through the glycolytic pathway, with pyruvate kinase highlighted as a pivotal control point. In healthy cells, the diagram would show pyruvate kinase, likely the PK-MON isoform, efficiently channeling pyruvate into the mitochondria for oxidative phosphorylation. However, in cancer cells, the focus would be on the PKM2 isoform, which can be less active, leading to a buildup of glycolytic intermediates, thus favoring biosynthetic pathways over complete glucose oxidation. This visual representation underscores how PKM2's regulation allows cancer cells to adapt their metabolism for growth, rather than just energy production. This brings us to the historical and research context, particularly the Warburg effect, named after Otto Warburg, who in the 1920s made groundbreaking observations on cancer metabolism. Warburg noted that cancer cells predominantly utilize glycolysis for energy production, even in the presence of oxygen, a metabolic trait that seemed paradoxical at the time. His work suggested that this shift towards aerobic glycolysis was not just an adaptation, but might be central to the cancer phenotype. Warburg hypothesized that this metabolic reprogramming could be a primary cause of cancer, rather than merely a consequence of genetic mutations, proposing that cancer cells have impaired mitochondrial respiration. Warburg's observations laid the groundwork for understanding cancer, not just as a disease of uncontrolled cell division, but as one deeply intertwined with metabolic alterations. His work has led to extensive research into how cancer cells rewire their metabolism, with pyruvate kinase, especially PKM2, becoming a focal point due to its regulatory role in this metabolic shift. This enzyme's ability to oscillate between states that either promote ATP production or the accumulation of biosynthetic precursors reflects the metabolic agility cancer cells require for survival and proliferation in hostile environments. Thus, pyruvate kinase stands at the crossroads of energy production and cell growth, embodying the metabolic dichotomy Warburg identified. The connection between Warburg's pioneering work, the catalytic action of pyruvate kinase, and modern diagrams of metabolic pathways paints a comprehensive picture of cancer metabolism. It illustrates how a fundamental biochemical reaction can have far-reaching implications for cancer biology, offering avenues for research into new cancer therapies by targeting metabolic vulnerabilities or by leveraging these differences for diagnostic purposes. The role of pyruvate kinase in metabolism is nuanced by the existence of its different isoforms, notably PKM-MON and PKM2, which have distinct expressions and functions. particularly in the context of cancer. PKMON is predominantly expressed in adult tissues like muscle and the brain, where it supports efficient ATP production through glycolysis and subsequent oxidative phosphorylation. On the other hand, PKM2 is found in embryonic tissues, proliferating cells, and notably in cancer cells, where it exhibits a unique regulatory behavior that favors tumor growth. The exclusive expression of PKM2 in tumor tissues is a hallmark of cancer metabolism. This isoform can exist in a less active dimeric form or a more active tetrameric form controlled by various cellular signals, including tyrosine phosphorylation, fructose 16, bisphosphate concentrations, and interaction with oncogenic proteins. This regulation allows cancer cells to adapt to their metabolism under varying conditions, particularly in hypoxic environments or when nutrients are scarce, by either promoting ATP production or diverting glycolytic intermediates into biosynthetic pathways for rapid cell division. Experimental evidence has shed light on the significance of isoform switching. Studies have demonstrated that replacing PKM2 with PKMON in cancer cells can have profound effects. This switch can lead to a reversal of the Warburg effect, where cancer cells shift back towards oxidative metabolism rather than glycolysis, reducing lactate production. This metabolic change can decrease the aggressive characteristics of cancer cells, as evidenced by reduced tumorigenicity, when these modified cells are implanted into xenograft models. The cells with PKMON show less capacity for rapid proliferation and less ability to form tumors, highlighting PKM2's role in supporting cancer's metabolic needs. Further metabolic studies comparing cells expressing PKMON versus PKM2 have elucidated these differences in detail. Oxygen consumption rates are higher in PKMON expressing cells, indicating a preference for oxidative phosphorylation, while PKM2 cells show increased lactate production, a direct result of aerobic glycolysis. Liquid chromatography mass spectrometry, LCMS, has been used to profile metabolites in these cells, revealing how PKM2's activity leads to the accumulation of glycolytic intermediates, which are then used for nucleotide amino acid and lipid synthesis, critical for cancer cell growth and survival. This body of research underscores the connection between pyruvate kinase isoforms and cancer's metabolic reprogramming. The switch to PKM2 in cancer cells is not merely an adaptation, but a strategic metabolic shift that supports the aggressive phenotype of cancer. It allows for the rapid generation of biomass for new cells, tolerance to hypoxic conditions, and potentially a mechanism to evade therapies targeting mitochondrial function. Understanding and possibly targeting this isoform switch could provide novel therapeutic strategies aiming to revert cancer cells to a less malignant metabolic state, or to exploit these metabolic state differences for diagnostic or treatment purposes. Thus, the study of pyruvate kinase isoforms not only illuminates cancer metabolism, but also offers a window into potential avenues for cancer control. Each PKM2, or pyruvate kinase M2 isoform, plays a pivotal role in the metabolic landscape of cancer cells, offering a selective advantage that supports tumor growth and survival. Studies have clearly demonstrated that when cancer cells predominantly express PKM2 rather than PKMN1, there's an observable increase in tumorigenic potential in vivo. This is largely because PKM2's unique biochemical properties allow cancer cells to adapt their metabolism to support rapid proliferation, even under conditions where resources are limited. The biochemical behavior of PKM2 is heavily influenced by its ability to undergo phosphorylation, which leads to significant conformational changes. When PKMA2 is phosphorylated on specific tyrosine residues, it tends to shift from an active tetrameric form to a less active dimeric state. This transformation reduces the enzyme's affinity for its substrate phosphoenolpyruvate, PEP, leading to a slowdown in pyruvate production and ATP generation. Instead, this change promotes the accumulation of glycolytic intermediates like glucose 6-phosphate and fructose 6-phosphate, which are then diverted into biosynthetic pathways. These intermediates are crucial for synthesizing nucleotides, amino acids, and lipids, which are necessary for the rapid division of cancer cells. This phosphorylation is not only a means of metabolic control, but also connects PKM2 to broader signaling pathways. PKM2 can bind to phosphotyrosine residues on other proteins, acting as a crossroads between glycolysis and cell signaling. This interaction can sequester PKM2 away from glycolytic activity, further supporting the accumulation of biosynthetic precursors. Further molecular insights into PKM2 reveal its complex regulation. PKM2 can form both tetramers and dimers, with the tetrameric form having a higher affinity for PEP, thus promoting glycolysis. However, the dimeric form has a lower affinity, which can be beneficial for cancer cells by allowing glycolytic intermediates to accumulate for other uses. Allosteric regulation by fructose 16-bisphosphate. FBP is another key aspect. FBP can convert PKM2 from the less active dimer to the highly active tetramer, providing a mechanism to rapidly switch between energy production and biosynthesis based on the cell's needs. The selective advantage PKM2 provides in cancer is multifaceted. By modulating its activity, cancer cells can survive and proliferate in environments where normal cells would struggle, such as those with low oxygen or nutrient levels. The ability of PKM2 to be regulated by phosphorylation and allosteric effectors, like FBP, allows for metabolic flexibility, enabling cancer cells to adapt to changing conditions within the tumor microenvironment. Moreover, the interaction of PKM2 with tyrosine phosphorylated proteins not only affects its enzymatic activity, but also positions PKM2 in the nucleus, where it can act as a protein kinase or transcription coactivator, influencing gene expression and further supporting cancer's aggressive phenotype. This intricate regulation and the connections between PKM2's biochemical characteristics, its role in metabolism, and its involvement in cellular signaling underscore why its expression is favored in cancer. Understanding these mechanisms offers potential targets for cancer therapy, where strategies might involve promoting PKM2's conversion to a tetramer to reduce the biosynthetic advantage of cancer cells, or targeting the pathways that lead to its phosphorylation, thereby aiming to disrupt the metabolic reprogramming central to cancer growth and survival. Pyruvate kinase M2. PKM2 is not only a gatekeeper of glycolysis, but also plays a crucial role in the de novo synthesis of serine, an amino acid vital for cancer cell proliferation. Serine's synthesis from glucose involves the diversion of glycolytic intermediates into the serine biosynthesis pathway, where PKM2's activity is pivotal. By controlling the rate of glycolysis, PKM2 can regulate the pool of 3-phosphoglycerate, a precursor for serine. This redirection supports the high demand for nucleotides, proteins, and other biomolecules in rapidly dividing cancer cells, showcasing PKM2's importance beyond just energy production. Interestingly, serine itself acts as an activator of PKM2. When serine levels are high, it binds to PKM2, promoting its transition to the tetrameric form, which increases its enzymatic activity. This activation can shift metabolic pathways from supporting biosynthetic needs back towards ATP production, demonstrating a feedback mechanism where serine availability can influence its own production rate and overall cellular metabolism. The integration of PKM2 into cellular signaling pathways further complicates its role. PKM2 activity is modulated by various cellular signals and intermediates, one notable example being SICAR, serine-induced complex for activating PKM2. SICAR is a complex that directly links serine levels to PKM2's activity, providing a regulatory loop where serine production can influence PKM2's tetramerization and thus its function in a metabolism. Moreover, PKM2 can be phosphorylated by kinases like SRC, leading to its dimerization and a decrease in glycolytic flux, favoring the accumulation of precursors for biosynthesis. This phosphorylation can be influenced by growth factors, oncogenic signals, or even cellular stress, showing PKM2 as a bridge between metabolic and signaling pathways. PKM2 also significantly influences the flow of carbon into the pentose phosphate pathway, PPPP. By controlling the availability of glucose 6-phosphate, PKM2 can direct metabolism towards the PPP, which is essential for producing NADF and ribose 5-phosphatase. NADF is crucial for maintaining redox balance and supporting biosynthetic reactions, while ribose 5-phosphate is needed for nucleotide synthesis. This diversion highlights how PKM2 can regulate not just energy production, but also the biosynthetic capacity of cells, particularly in cancer, where these pathways are ramped up. The Warburg effect, where cancer cells prefer aerobic glycolysis despite oxygen availability, seems inefficient at first glance. However, this metabolic shift might confer several selective advantages. One speculation is that by utilizing glycolysis, cancer cells can generate ATP rapidly, without relying on oxygen, which might be scarce in densely packed tumors. Moreover, this less efficient metabolism allows for the accumulation of glycolytic intermediates, supporting rapid biomass production. The acidic environment, created by lactate production, can inhibit the activity of normal cells and immune responses, creating a niche favorable for cancer growth. Additionally, this metabolic reprogramming might reduce the generation of reactive oxygen species, ROS, that would otherwise damage DNA, providing a protective mechanism against oxidative stress. Thus, PKM2's role in cancer is emblematic of how metabolic pathways are not just about energy, but are intricately linked with signaling, biosynthetic demands, and survival strategies. Understanding these connections offers insights into why cancer cells adopt seemingly counterintuitive metabolic behaviors and suggests potential therapeutic targets aimed at disrupting this metabolic advantage. Lecture 5. The tumor microenvironment, TEAM, is a complex and dynamic ecosystem that profoundly influences cancer progression, treatment response, and resistance. It comprises not just the cancer cells themselves, but also a variety of other cell types, including immune cells, cancer-associated fibroblasts, and endothelial cells, all embedded within an altered extracellular matrix, EMM. This milieu is far from static. It's a living, breathing environment where interactions between its components can either promote or inhibit tumor growth, metastasis, and immune evasion. The cellular landscape of the TEAM includes tumor cells, which are genetically and epigenetically altered to thrive in this environment. Surrounding them, immune cells like tumor-associated macrophages, TAMs, T cells, and B cells, often co-opted by the tumor to support its growth rather than fight it, create an immunosuppressive environment. Cancer-associated fibroblasts, CAFs, are another key player, remodeling the ECM to facilitate invasion and providing growth factors that support tumor expansion. The ECM itself, with its altered composition, creates physical barriers to drug delivery and immune cell infiltration, while also providing cues that can drive cancer cell behavior. The physicochemical properties of the TEAM are equally telling of its role in cancer biology. One of the most striking features is the existence of metabolic gradients across the tumor. As one moves from the well-vascularized periphery towards the hypoxic core of a tumor, there are significant changes in pH, oxygen availability, nutrient concentrations, and the accumulation of metabolic byproducts like lactate. Hypoxia, or low oxygen tension, is a hallmark of many tumors due to their rapid growth outpacing the development of an adequate blood supply. This hypoxia triggers a cascade of responses, including the stabilization of hypoxia-inducible factors, HIFs, which in turn promote angiogenesis, metabolic reprogramming towards glycolysis, and survival mechanisms against oxygen deprivation. The shift towards glycolysis, even in the presence of oxygen, the Warburg effect, results in the production of lactate, leading to an acidic microenvironment. This acidity, with pH often lower than in normal tissues, not only affects the function of surrounding cells, including immune cells, but also alters the ECM, promoting invasion and metastasis. The acidic and hypoxic time has profound implications. Acidity can deactivate many chemotherapeutic drugs, making cancers more resistant to treatment. It can also impair immune cell function, particularly T cell activity, thereby helping cancer cells evade immune surveillance. Hypoxia, on the other hand, drives genetic instability, fostering further mutations that could lead to increased malignancy and resistance. It also induces changes in cellular signaling, promoting survival under stress invasion and angiogenesis. These physicochemical conditions within the TE are not merely consequences of tumor growth, but actively contribute to cancer progression. They create selective pressures that might favor cells with specific metabolic or genetic adaptations, leading to the evolution of more aggressive cancer phenotypes. Furthermore, the metabolic stress within the time can instruct normal cells to adopt pro-tumorigenic roles, further sculpting an environment conducive to cancer survival and spread. Understanding the tumor microenvironment, therefore, isn't just about recognizing its components, but also about grasping how these physicochemical properties shape cancer biology. This understanding opens avenues for therapeutic strategies that target not just the cancer cells, but also the supportive stroma metabolic adaptations, or the physical barriers within the time, aiming to disrupt the cancer's home advantage and enhance treatment efficacy. Lactate, traditionally seen as a byproduct of glycolysis, has emerged as a significant player in the tumor microenvironment, TINE, influencing both the metabolic and immunological landscape of cancer. The production of lactate by cancer cells, particularly through the Warburg effect, leads to its accumulation in the TIME, creating an acidic environment that has profound implications for tumor biology. One of the fascinating aspects of lactate in the time is its role in establishing a metabolic symbiosis between cancer cells and the surrounding stroma. This symbiosis is facilitated by monocarboxylate transporters, MCTs, specifically M-ketone and MT4. Cancer cells that are highly glycolytic export lactate via MCT4, which then can be taken up by neighboring stromal cells, including cancer-associated fibroblasts, CAFEs, and certain immune cells. Through mTOR1, these cells can utilize lactate as an energy source, converting it back into pyruvate for oxidative phosphorylation in their mitochondria. This metabolic crosstalk creates a supportive environment where stroma cells indirectly fuel cancer cell proliferation by recycling lactate, thus enhancing the tumor's metabolic efficiency and growth. Beyond this metabolic symbiosis, lactate significantly contributes to immune suppression within the TIME. The acidic environment created by lactate accumulation impairs the function of various immune cells. For T cells, the low pH can inhibit their proliferation, cytokine production, and cytotoxic activity, essentially blunting the immune response against the tumor. Lactate also affects dendritic cell activation, which are key in antigen presentation and initiating immune responses. In an acidic environment, dendritic cells tend to have reduced capacity for antigen presentation, skewing the immune system towards tolerance rather than an anti-tumor response. Furthermore, lactate influences the polarization of macrophages and the function of myeloid-derived suppressor cells, MDSCs. High lactate levels can shift macrophage polarization towards an M2-like phenotype, which is associated with promoting tumor growth, angiogenesis, and immune suppression, rather than fighting the cancer. Similarly, MDSCAs, which are known for their ability to suppress T-cell activity, can be further empowered by the lactate-rich environment, exacerbating immune evasion by cancer. This multifaceted role of lactate in the thyme illustrates how cancer cells can manipulate their surroundings, not just for their own metabolic advantage, but also to create an immunologically permissive environment. The production and export of lactate by cancer cells thus serve dual purposes, supporting the tumor's growth through metabolic symbiosis and shielding it from immune attack through suppression mechanisms. Understanding these connections between lactate and the thyme offers potential therapeutic avenues. Strategies might include targeting MCT transporters to disrupt the lactate shuttle, altering the acidic environment to restore immune function, or even exploiting these metabolic vulnerabilities for selective cancer cell killing. Such approaches aim at not only directly combating cancer cells, but also at reprogramming the TME to make it less hospitable for cancer growth and more conducive to immune-mediated tumor clearance. Thus, the study of lactate in cancer biology unveils a layer of complexity where metabolism and immunity are intricately linked, highlighting novel targets for intervention in the fight against cancer. In the context of prostate cancer, the interplay between lactate and lipid metabolism reveals a sophisticated mechanism of tumor sustenance and growth, mediated significantly by cancer-associated fibroblasts, CAFs. CAFs within the tumor microenvironment, TEE, actively participate in this metabolic crosstalk, utilizing the lactate exported by cancer cells to fuel their own metabolic processes, which in turn supports cancer cell proliferation. CAFs take up lactate via mectone transporters, converting it back into pyruvate, which can be metabolized through the TCA cycle in the mitochondria. This conversion not only serves to recycle the lactate, but also generates intermediates like citrate. Citrate is particularly crucial as it can be shuttled back to the cytosol where it's cleaved into acetyl-CoA and oxaloacetate by ATP citrate lyase. Acetyl-CoA is then a primary building block for de novo lipogenesis, the synthesis of fatty acids necessary for membrane biogenesis, energy storage, and signaling molecules in cancer cells. Thus, CAFs, by metabolizing lactate, indirectly enhance lipid synthesis in cancer cells, providing them with the lipids they need for rapid growth and division. The influence of lactate doesn't stop at metabolite exchange. It extends to gene expression levels. Lactate has been shown to impact the expression of genes involved in lipid metabolism within prostate cancer cells. For instance, high lactate concentrations can induce the expression of key lipogenic genes like fatty acid synthase, FASN, and sterol regulatory element binding protein, 1, SREBPON, both of which are pivotal in the synthesis of fatty acids and cholesterol. Through these transcriptional changes, lactate can reprogram the cancer cell's metabolic state towards increased lipid production, further supporting the aggressive phenotype of prostate cancer. Experimental methodologies have provided concrete evidence of these phenomena. Conditioned media experiments where media from CAFs is used to culture prostate cancer cells have demonstrated that the presence of lactate in this media can significantly upregulate lipid metabolism in cancer cells. This approach allows researchers to observe how the metabolic products of one cell type can influence another in the timeline. Isotopy tracing studies using labeled lactate, such as 14C or 13C, or 13C lactate, have been instrumental in tracing the metabolic fate of lactate within cells. These experiments have shown that lactate derived from CAFs does indeed contribute to the pool of carbon used in lipid biosynthesis. By following the labeled carbon through the metabolic pathways, researchers can ascertain the extent to which lactate is converted into various lipid precursors, providing direct evidence of lactate's role in supporting lipid synthesis in prostate cancer cells. These findings highlight not just the metabolic versatility of cancer cells, but also the interconnectedness of different cell types within the NNEN. The lactate lipid axis in prostate cancer exemplifies how cancer cells can leverage their environment for growth, showcasing the potential of targeting these metabolic interactions for therapeutic intervention. By disrupting lactate uptake or utilization, or by modulating the lipid synthesis pathways activated by lactate, there might be opportunities to slow down cancer progression or enhance the efficacy of existing treatments. This research thus opens up new avenues for understanding cancer biology and for developing strategies that could exploit these metabolic dependencies to the advantage of cancer treatment. Lipid droplets, LDS, are organelles that serve as dynamic storage depots for neutral lipids, like triglycerides and cholesterol esters within cells. In the context of cancer, particularly in cells exposed to lactate derived from cancer-associated fibroblasts, there is a notable accumulation of these lipid droplets. This phenomenon is not merely a byproduct of altered metabolism, but has significant functional implications for cancer cell biology. The increase in LDS within cancer cells can be seen as a direct consequence of the metabolic symbiosis facilitated by lactatia. When calves metabolize lactate into pyruvate, which then enters the TCA cycle, the resulting citrate can be shuttled out of the mitochondria for cytosolic lipid synthesis. This process leads to an accumulation of lipids which are then stored in LDS. These LDS are not just passive storage units. They actively participate in the metabolic flexibility of cancer cells, providing a quick access reservoir of energy and structural components. The functional significance of LDS in cancer cells extends beyond mere storage. They act as an energy reserve, particularly under conditions of nutrient stress or hypoxia, where cancer cells might need to switch from glycolysis to oxidative metabolism. LDS can be mobilized to release fatty acids, which can then be oxidized in mitochondria to produce ATP through beta oxidation, thus supporting the cell's energy needs when glucose is scarce or when the Warburg effect is not advantageous. This adaptation allows cancer cells to survive and proliferate in an environment where metabolic conditions fluctuate. Moreover, LDS are central to cholesterol homeostasis within cancer cells. Cholesterol is vital for membrane synthesis, signal transduction, and maintaining cellular integrity, all of which are heightened in rapidly dividing cancer cells. LDS store cholesterol esters, which can be hydrolyzed to provide free cholesterol when needed, supporting membrane biogenesis for new cells. This is particularly relevant in cancers like prostate cancer, where lipid metabolism is significantly upregulated. The presence of increased LDS in cancer cells also has implications for resistance to therapies. Cancer cells can use lipids from LDS to counteract oxidative stress, a common mechanism of action for many chemotherapeutic agents. By providing substrates for antioxidant pathways, or by altering membrane fluidity, and thus affecting drug uptake, LDS can contribute to a survival advantage for cancer cells. This accumulation and utilization of LDS highlight a broader theme in cancer biology: the adaptation and exploitation of cellular metabolic pathways to support uncontrolled growth and survival. The connection between calf-derived lactate and LD accumulation underscores the complexity of the tumor microenvironment, where different cell types engage in metabolic crosstalk to foster cancer progression. Understanding this interplay not only sheds light on cancer's metabolic reprogramming, but also suggests novel therapeutic targets. Strategies might include disrupting the formation or utilization of LDS, or targeting the pathways through which lactate influences lipid metabolism, aiming to deprive cancer cells of these critical energy and structural resources. Metabolic reprogramming is a hallmark of cancer, where cells adjust their metabolic pathways to support rapid growth and survival under the challenging conditions of the tumor microenvironment. Central to this reprogramming in the presence of lactate is the upregulation of enzymes involved in lipid anabolism, notably ATP, citrate, lyase, acly, and acetyl-CoA, carboxylase, ACC, show increased expression levels when cancer cells are exposed to high lactate environments. OI converts citrate from the TCA cycle into acetyl-CoA and oxaloacetate, providing the building blocks for fatty acid synthesis. EC then catalyzes the carboxylation of acetyl-CoA to malonyl-CoA, the first committed step in de novo fatty acid synthesis. This enzymatic cascade, spurred by lactate, drives the synthesis of lipids, contributing to the formation and accumulation of lipid droplets, LDS. The therapeutic implications of this metabolic shift towards lipid anabolism and LDL accumulation are profound. One potential strategy involves targeting the release of lipids from LDS, thereby impacting the oxidative capacity of cancer cells. When lipids are sequestered within LDS, they are not immediately available for energy metabolism. By inhibiting their mobilization, cancer cells might be forced into a state of energy deprivation or oxidative stress, particularly under conditions where they rely on lipid oxidation for survival, such as in hypoxic or nutrient-poor environments. Inhibition studies have provided insights into LDS role in cancer cell metabolism. Compounds like aglystatin, which inhibits adipose triglyceride lipase, AGL, the enzyme responsible for catalyzing the first step in triglyceride hydrolysis from LDS, have been used to demonstrate the dependency of cancer cells on lipid mobilization for energy. By blocking aglystatin can prevent the release of fatty acids, potentially starving cancer cells of energy when they switch to beta oxidation for ATP production. Similarly, etomoxir, an inhibitor of carnitine palmitoyl-transferase 1, CP-tone, which is crucial for transporting fatty acids into mitochondria for oxidation, has been employed to study the impact of disrupting lipid catabolism on cancer cell viability. These inhibitors highlight how targeting lipid metabolism, specifically the dynamics of LDS, can be a viable strategy for cancer treatment. The connection between lactate enzyme upregulation, LD accumulation, and the potential for therapeutic intervention paints a picture of cancer as a disease deeply rooted in metabolic dysregulation. By understanding and targeting these metabolic pathways, particularly those involving lipid metabolism, researchers can aim to disrupt cancer cells' ability to adapt to their harsh microenvironment. Such strategies not only aim at reducing cancer cell proliferation and survival, but also at enhancing the effectiveness of existing treatments by making cancer cells more susceptible to oxidative stress or by limiting their metabolic flexibility. This approach underscores the importance of metabolic reprogramming in cancer biology, offering a pathway to novel treatments that focus on the unique metabolic vulnerabilities of cancer cells. Lecture 6. Metabolomics is a dynamic field within the broader omics sciences that focuses on the study of small molecules, known as metabolites, which are the end products of cellular processes. These molecules serve as functional readouts of cellular and molecular programs, providing a direct snapshot of the physiological state of an organism or cell at any given moment. By analyzing the levels, types, and interactions of these metabolites, researchers can gain insights into metabolic pathways, disease mechanisms, and the effects of genetic or environmental changes on metabolism. The applications of metabolomics are vast and impactful. One of the primary uses is in biomarker discovery, where unique metabolite profiles can indicate the presence of a disease before traditional symptoms manifest, offering potential for early diagnosis. In disease metabolomics, comparing metabolite profiles from healthy versus disease states can elucidate the metabolic perturbations associated with conditions like cancer, diabetes, or cardiovascular diseases, leading to a better understanding of disease pathology and progression. This knowledge also paves the way for personalized medicine where treatment can be tailored based on an individual's unique metabolic signature, potentially improving efficacy and reducing side effects. In terms of methodologies, metabolomics can be approached through several techniques, each with its advantages. Bulk metabolomics involves analyzing the metabolite profile of a large sample, providing an overview of metabolic changes but lacking spatial resolution. Partial and single-cell metabolomics, on the other hand, offer insights into metabolic heterogeneity within tissues or between cells, crucial for understanding complex diseases like cancer, where different regions or cell types might behave metabolically differently. Isotopic tracing is another powerful tool where labeled substrates like 13C or 15 are introduced to track metabolic pathways, revealing how nutrients are processed and utilized within cells. However, metabolomics faces several challenges. Achieving high accuracy and resolution in metabolite identification and quantification is paramount, but technically demanding. The dynamic range of metabolite concentrations in biological samples is vast, necessitating instruments with exceptional sensitivity to detect both highly abundant and trace metabolite. Furthermore, the complexity of biological samples introduces issues of matrix effects, where the sample's composition can interfere with metabolite detection or quantification. The integration of metabolomics data with other omics data, genomics, transcriptomics, proteomics, provides a holistic view of biological systems, highlighting how genetic and environmental factors translate into metabolic phenotypes. This integrative approach is key to understanding complex diseases where multiple layers of biological regulation are at play. The connections between metabolomics and health sciences are profound. By revealing the metabolic underpinnings of disease, metabolomics not only aids in diagnosis and treatment, but also in understanding the basic biology of life. It's a testament to the interconnectedness of all biological processes, where a change at the genetic level can manifest through metabolic changes influencing health outcomes. As technology advances, particularly in terms of sensitivity resolution and the ability to perform non-invasive or in vivo measurements, metabolomics will continue to revolutionize our approach to medicine, from basic research to clinical applications, making it a cornerstone of modern biological and medical science. Metabolites represent an incredibly diverse group of small molecules within biological systems, encompassing everything from endogenous compounds produced by the body or cell to xenobiotics, which are foreign chemicals from the environment, drugs, or diet. This diversity extends to include microbial metabolites, which are produced by the vast microbiome within humans and other organisms, influencing health and disease. Plant metabolites, critical in understanding plant biology and pharmacology, and human metabolites, which are central to our understanding of human physiology, pathology, and pharmacology. Estimates of the number of known metabolites vary by species, but are vast. For humans, there are tens of thousands of identified metabolites, each potentially playing unique roles in cellular function signaling and disease states. This number grows when considering the metabolites from our gut microbiome, which can produce thousands more. In plants, the metabolite diversity is even greater, with estimates suggesting hundreds of thousands of different compounds, many of which contribute to the plant's defense, growth, and interaction with the environment. The history and evolution of metabolomics trace back to the 1980s, initially with the use of nuclear magnetic resonance, NMR, spectroscopy for metabolite identification. NMR provided a non-destructive way to analyze complex mixtures of small molecules in biological samples, offering insights into metabolic states. However, the field truly expanded with the advent of mass spectrometry, MES techniques in the 1990s and beyond. This especially when coupled with liquid chromatography, LCMs, or gas chromatography, GCMs, dramatically increased the sensitivity, specificity, and throughput of metabolite detection, allowing for the analysis of thousands of metabolites in a single experiment. This technological evolution has been accompanied by a surge in scientific literature with over 17 million publications now dedicated to the study of metabolites and lipids. This body of work spans from foundational research on metabolic pathways to applied studies in biomarker discovery for diseases like cancer, diabetes, and neurodegenerative disorders. The literature not only documents the expansion from metabolite identification to understanding their roles in complex biological systems, but also charts the path from basic research to clinical applications. The connections between the diversity of metabolites and the evolution of metabolomics research are profound. Each technological advance has expanded our capacity to explore this diversity, moving from broad, less detailed pictures of metabolism to high-resolution maps where individual metabolic pathways can be traced and their perturbations in disease understood. This has implications for nearly every field of biology and medicine, from pharmacology, where understanding drug metabolism is crucial, to nutrition, where the metabolic impact of diet can be precisely characterized, to environmental science, where the effects of xenobiotics on organisms can be studied. As metabolomics continues to evolve, with techniques becoming more sensitive, integrative, and able to handle complex biological matrices, it promises to further illuminate the intricate web of life at the molecular level. This not only advances our understanding of health and disease, but also drives innovation in drug discovery, personalized medicine, and environmental monitoring, all underpinned by the fundamental principle that our biochemistry is a direct reflection of our health, environment, and lifestyle. In the realm of metabolomics, two primary approaches define the analytical landscape: untargeted and targeted metabolomics. Untargeted metabolomics aims to cast a wide net, capturing a comprehensive profile of all detectable metabolites within a sample. This method is particularly useful for hypothesis generation, allowing researchers to identify unexpected metabolic changes, discover new biomarkers, or understand complex biological systems without preconceived notions about which metabolites are of interest. It provides a broad snapshot of the metabolic state, but can be challenging due to the sheer volume of data generated and the need for sophisticated computational tools to interpret it. On the other hand, targeted metabolomics focuses on specific metabolites or metabolic pathways known or suspected to be relevant to a particular study or disease. This approach is more quantitative, offering precise measurements of selected metabolites, which is crucial for validating hypotheses from untargeted studies, monitoring disease progression, or evaluating the effects of interventions like drug treatments. It's less about discovery and more about confirmation and detailed characterization. Tools and methods in metabolomics vary based on the scale and detail of analysis required. Bulk analysis is the standard for metabolomics, where large sample sets are processed to understand general metabolic trends or to identify biomarkers across a population. Techniques like LCMs, liquid chromatography mass spectrometry, and GCMs, gas chromatography mass spectrometry, dominate this field, providing high-throughput analysis, but at the cost of spatial resolution and individual cell insights. However, the field is advancing into more nuanced techniques like single-cell metabolomics, which offer unprecedented detail. For instance, Nimi's lab has pioneered the use of microfabricated capillaries for analyzing metabolites in individual xenopus embryos, allowing the study of metabolic variations at the earliest stages of life and development. This method not only increases sensitivity, but also provides insights into cellular heterogeneity, crucial for understanding diseases like cancer, where metabolic profiles can vary dramatically from cell to cell. Sweedler's lab has taken an innovative approach with image-guided multi-Feker, matrix-assisted laser desorption, ionization Fourier transform, ion cyclotron resonance, which allows for the direct analysis of metabolites from specific vesicles within cells. This technique combines high-resolution imaging with mass spectrometry to map metabolites, spatially offering a glimpse into how individual cellular compartments contribute to overall metabolism. Another groundbreaking method comes from Xiang's lab, which uses lysosomal patch clamp combined with NanoACM's nanoelectrospray ionization mass spectrometry to explore the metabolism of lysosomes in single cells. This approach not only sheds light on the metabolic activity within these organelles, but also connects cellular metabolism with lysosomal function, potentially revealing new aspects of metabolic regulation and disease mechanisms. These advanced techniques highlight the interconnectivity between metabolomics and cellular biology, showing how metabolic profiling can be leveraged to understand not just bulk tissue responses, but just bulk. Also, the nuanced cell-to-cell differences that drive biological processes. Each method, from bulk to single-cell analysis, contributes to a more comprehensive understanding of how metabolites influence health, disease, and the response to therapeutic interventions, bridging the gap between molecular biology, biochemistry, and clinical practice. Spatial metabolomics, particularly through mass spectrometry imaging, MSI, offers a groundbreaking approach to visualize the distribution of metabolites directly within biological tissues, providing insights into how metabolic processes occur spatially within organs or across disease states. This technique involves taking thin sections of tissues, which are then sampled and ionized to map the presence and concentration of various metabolites. The process generally starts with the preparation of the tissue, often involving fixation and sectioning to very thin slices, which are then mounted onto slides. The choice of ionization modality can vary with techniques like MALDI, matrix-assisted laser desorption ionization, using a laser to ionize metabolites after they've been co-crystallized with a matrix. Other methods include SIMS, secondary ion mass spectrometry, which uses an ion beam, or DESI, desorption electrospray ionization, involving a spray of charged solvent. Each modality has its advantages, with MALDI being widely used for its ability to analyze a broad range of biomolecules with good spatial resolution. An example of this technology in action is the use of the MALDI timsTOF Pro for imaging the metabolome of a rat kidney. This system, combining MALDI with time-of-flight mass spectrometry, provides detailed maps of metabolite distribution across different kidney regions, like the cortex, medulla, and papilla. Such imaging can reveal how kidney function, particularly in terms of metabolite handling, varies spatially, offering insights into how different parts of the organ might respond to physiological keys, changes, or disease states. In terms of disease study, diabetic nephropathy serves as an illustrative case. This condition, a complication of diabetes, involves significant metabolic alterations in the kidney, particularly within the glomeruli. Hyperglycemia leads to changes in the glomerulus metabolome, where the accumulation of glucose and its derivatives can alter lipid metabolism, leading to the production of modified lipids that contribute to inflammation and fibrosis. Using MALDI FDKR, MS, Fourier transform, ion cyclotron resonance mass spectrometry, researchers can detect these changes at high resolution. This technique not only identifies the modified lipids, but also maps their distribution across the glomerulus, showing where these metabolic shifts are most pronounced and potentially linking these changes to specific pathological features like glomerular sclerosis or loss of filtration capacity. The connections here are multifaceted. On one hand, spatial metabolomics provides a direct link between metabolic changes and anatomical features, allowing for a more nuanced understanding of how diseases like diabetic nephropathy develop at the molecular level. On the other hand, by visualizing these changes, researchers can identify potential biomarkers or therapeutic targets, which are spatially specific. For instance, targeting lipid accumulation in certain parts of the glomerulus to prevent progression to end-stage renal disease. This approach not only enhances our basic understanding of disease pathology, but also has practical implications for diagnosis, monitoring, and treatment. By recognizing that metabolic changes in diseases are not uniform across tissues, but rather localized, we can aim for therapies that are more targeted, potentially reducing side effects and improving efficacy. Thus, spatial metabolomics stands as a bridge between the molecular intricacies of metabolism and the macroscopic outcomes of disease, providing a rich canvas for both research and clinical application. The future of metabolomics is set to expand our understanding of biological systems through increasingly sophisticated techniques. With single-cell metabolomics, SMetabolomics, at the forefront of this evolution. This approach seeks to unravel the metabolic heterogeneity that exists within tissues, or even within different parts of the same cell, offering insights into how individual cells contribute to overall tissue function or disease pathology. One promising direction involves integrating metabolomics with Raman microscopy. Raman microscopy uses laser light to scatter off molecules in a way that provides a unique molecular fingerprint of the compound's presentile. By combining this with single-cell metabolomics, researchers can achieve non-destructive, real-time imaging of metabolites within individual cells. This synergy could revolutionize our understanding of dynamic metabolic processes, providing a live view of how cells respond to environmental changes, drug treatments, or disease progression. Such integration not only enhances the spatial and temporal resolution of metabolic studies, but also allows for the simultaneous assessment of metabolite distribution and cellular morphology or function, offering a more holistic view of cellular biology. Another avenue for advancement is the expansion of nanodesi, desorption electrospray ionization at the nanoscale, applications. Originally developed for analyzing surface chemistry with minimal sample preparation, nanodesi can now be adapted to a wide range of biological samples, from plant tissues to human biopsies. This technique allows for the sampling of metabolites directly from the surface of biological materials, providing high-resolution spatial information about metabolite distribution. By extending nanodesi to various biological contexts, researchers can explore the microenvironments within tissues, such as the tumor microenvironment, or the interface between plant roots and soil. Microorganisms yielding insights into how these interactions influence metabolism at a very local level. Improving spatial resolution and metabolite coverage is also a critical focus. Current technologies in metabolomics are constantly being refined to increase the detail and breadth of metabolite detection. Advances in mass spectrometry, such as higher resolution, mass analyzers, or new ionization methods, are expected to push the boundaries, allowing for the detection of a wider array of metabolites with finer spatial detail. This improvement is crucial for mapping complex metabolic networks, where the location of a metabolite can be as informative as its presence or concentration, particularly in understanding diseases where metabolic changes are spatially heterogeneous. The connections between these future directions are profound. Each technological leap in metabolomics, whether through integration with Raman microscopy, expanding the use of nanodesi, or enhancing mass spectrometry's capabilities, not only increases our ability to detect and analyze metabolites, but also redefines our approach to biological research. By understanding metabolism at the single-cell level, we can better grasp how cellular diversity contributes to organ function or disease. This knowledge is pivotal for personalized medicine, where treatments can be tailored to the specific metabolic profiles of an individual's cells, potentially leading to more effective therapies with reduced side effects. Furthermore, These advancements connect to broader scientific themes, such as the interplay between technology and discovery, where each new tool not only answers existing questions, but also illuminates new ones to explore. As we continue to refine these technologies, we are not just looking at a future where we understand more about metabolism. We are envisioning a new era where our approach to biology, medicine, and even environmental science is fundamentally reshaped by the insights gained from these microscopic metabolic landscapes. Lecture 7. Lipid metabolism in cancer represents a critical aspect of tumor biology, where cancer cells exploit both intrinsic and extrinsic sources of lipids to fuel their rapid proliferation, invasion, and survival. This metabolic reprogramming involves several key enzymes and pathways that are often upregulated or dysregulated in cancer cells. Starting with the key enzymes, ATP, citrate lyase, ACLY, plays an essential role by converting citrate, a product of the TCA cycle, into acetyl-CoA in the cytosol in the cytosol. This substrate is fundamental for both lipid and cholesterol synthesis. Acetyl-CoA carboxylase, ACSY, then catalyzes the carboxylation of acetyl-CoA to malonyl-CoA, the first committed step in de novo fatty acid synthesis. Fatty acid synthase further extends this process by synthesizing long-chain fatty acids from malonyl-CoA and acetyl-CoA. Another pivotal enzyme, hydroxymethylglutaryl-CoA reductase, AMGCR, is central to cholesterol biosynthesis, converting NGMG-CoA to mevalonate, which is a rate-limiting step in the mevalonate pathway. The intrinsic supply of lipids in cancer cells primarily comes from de novo lipogenesis, where cancer cells produce their own fatty acids and cholesterol to meet the high demand for membrane synthesis, signaling molecules, and energy storage. This process is particularly pronounced in cancers, where external lipid supply might be limited, or where the tumor microenvironment is hypoxic or nutrient poor. De novo lipogenesis is heavily influenced by the activity of ACLY-ACC phasin and the transcriptional regulation by sterol regulatory element binding proteins, SREPs, which are master regulators of lipid metabolism. Extrinsic lipid supply involves the uptake of lipids from the tumor microenvironment or from circulation. Cancer cells can increase their lipid uptake through various mechanisms, including the upregulation of fatty acid binding proteins, FAPs, which transport fatty acids into cells, and the modification of lipid receptors or transporters on the cell surface. This external supply is crucial in environments where tumor cells can efficiently scavenge lipids from the surroundings, such as from adipocytes in the vicinity of tumors or through lipoprotein uptake from the bloodstream. Fatty acid oxidation, FAO, represents another facet of lipid metabolism in cancer, particularly in situations where glucose is scarce, or when cells switch from glycolysis to oxidative metabolism. For energy, enzymes like carnitine palmitoyltransferase 1, CP-tone, are critical for transporting fatty acids into mitochondria for oxidation. Acyl-CoA synthetase, short-chain family member 1, axon 1, activates acetate for mitochondrial entry, while acetyl-CoA acetyltransferase, ACAT, aids in the synthesis of cholesterol esters for 4-esters 4. Storage in lipid droplets. Steroyl-CoA desaturase, SAD, also plays a role by introducing double bonds into fatty acids, affecting their fluidity and function in membrane. The connections between these pathways and enzymes are intricate. Cancer cells can toggle between using lipids for energy or biosynthesis based on the availability of nutrients or oxygen, showcasing their metabolic flexibility. This adaptability is not just about survival, but about supporting aggressive cancer behaviors like metastasis, where lipids are needed for new membrane synthesis, or a signaling molecule. Furthermore, the upregulation of these pathways can lead to the accumulation of lipid droplets, serving as a reservoir of energy and biomaterials, which cancer cells can utilize under stress conditions. Understanding lipid metabolism in cancer opens up avenues for therapeutic intervention, where targeting key enzymes like FASN or AMGKR or modulating lipid uptake or FOAM might starve cancer cells of essential building blocks or energy, potentially slowing down tumor growth or sensitizing them to other treatments. This biochemical landscape thus not only explains how cancer cells manage to thrive, but also illuminates potential strategies for cancer control through metabolic targeting. Cancer-associated fibroblasts, CAFs, play a pivotal role in the tumor microenvironment, not only through structural support, but also by influencing cancer cell metabolism, particularly in lipid anabolism. One of the key mechanisms through which CAFs support cancer cell growth is via the export of lactate, which becomes a substrate for lipid synthesis in cancer cells. Lactate produced in high amounts by CAFs due to their glycolytic metabolism is taken up by cancer cells where it's converted back into pyruvate. This pyruvate can then enter the TCA cycle, producing citrate, which is subsequently exported to the cytosol. Here, ATPP, citrate lyase, converts citrate into acetyl-CoA, a building block for lipid synthesis. This process leads to the accumulation of lipid droplets, LDS, organelles that store neutral lipids like triglycerides and cholesterol esters, serving as dynamic reserves of energy and structural components for rapidly dividing cancer cells. In prostate cancer, for instance, exposure to CAF-conditioned media, CAF-Ciniacte, has been shown to significantly increase the number and size of LDS. These LDS act not just as storage, but as a source of lipids for membrane synthesis, signaling molecules and a buffer against oxidative stress. The presence of increased LDS in cancer cells exposed to this lactate environment underscores a metabolic symbiosis, where CAFs indirectly fuel cancer cell proliferation by providing the raw materials for lipid synthesis. The impact of inhibitors further elucidates this relationship. Inhibitors of monocarboxylate transporter 1, NIM-ketone, which CAFs and cancer cells use to import and export lactate, can reduce the uptake of lactate by cancer cells, thereby decreasing old formation. Similarly, inhibiting ACLY, or AMG-CoA reductase, AMG-CR, directly affects lipid anabolism. By blocking ACLY, the conversion of citrate to lipid precursors is curtailed, while AMG-CR inhibition disrupts cholesterol synthesis, both of which are critical for LD formation. These interventions demonstrate that targeting the pathways through which lactate influences lipid metabolism can potentially slow down cancer cell proliferation by limiting their lipid supply. The energetic role of LDS in cancer cells is highlighted by experiments involving oxygen consumption rate, OCR. LDS are not static. They are mobilized to provide fatty acids for beta oxidation in mitochondria, particularly under conditions where glucose is scarce or when cells need to switch from glycolysis to oxidative metabolism for energy. When aid lipid release is inhibited using compounds like adglistatin, which blocks adipose triglyceride lipase, ADGL, the OCR decreases, indicating that the lipids stored in LDS are indeed a significant source of energy through fatty acid oxidation. This confirms that LDS serve dual roles, not just in biosynthesis, but also as an energy reservoir allowing cancer cells metabolic flexibility to survive in fluctuating conditions within the TEEN. The connections here weave a complex narrative where the metabolic interdependence between CAFs and cancer cells, mediated by lactate, directly impacts lipid metabolism, cell growth, and survival strategies. Understanding and potentially disrupting this metabolic crosstalk could offer new therapeutic avenues aiming at both the cancer cells and the supportive stroma, leveraging metabolic vulnerabilities to halt cancer progression. Fatty oxidation. FO is a critical metabolic pathway in cells, particularly in cancer, where it serves as an alternative energy source when glucose is limited or when cells shift towards oxidative metabolism. This process begins with the transport of long-chain fatty acids into the mitochondria, the powerhouse of the cell where these fatty acids are ultimately oxidized to generate ATP. The initial step in FAO involves the activation of fatty acids to their co-esters, acyl-CoA, in the cytoplasm. These acyl-CoAs are then ferried across the mitochondrial membranes through a shuttle system involving carnitine. Here, carnitine palmitoyl transferase I, CP-tone, located on the outer mitochondrial membrane, catalyzes the conversion of acyl-CoA to acylcarnitine, allowing it to pass through the inner membrane via the carnitine-acyl-carnitine translocation. Once inside, carnitine palmitoyl transferase, CPD, reverses this process, converting acylcarnitine back to acyl-CoA for entry into the beta-oxidation pathway within the mitochondrial matrix. Beta-oxidation sequentially cleaves 2 carbon units from the fatty acid, producing acetyl-CoA, which then enters the TCA cycle for further oxidation, or can be used for ketone body synthesis or other biosynthetic pathways. The link between FAO and epigenetics, particularly through acetyl-CoA, underscores the deep interconnection between metabolism and gene regulation. Acetyl-CoA, a product of FAO, is not only an energy metabolite, but also a crucial substrate for histone acetylation, which affects gene expression by altering chromatin structure. In cancer, the reprogramming of lipid metabolism, often influenced by lactate from the tumor microenvironment, can lead to changes in the availability of acetyl-CoA. This, in turn, impacts histone acetylation patterns, potentially activating genes that favor cancer progression, such as those involved in proliferation, survival, and metastasis. Thus, the metabolic state of a cell can directly influence its epigenetic landscape, demonstrating how cancer cells can adapt both their metabolism and gene expression to support aggressive phenotypes. The impact of this metabolic epigenetic interaction extends to cancer cell invasiveness. By reducing lipogenesis, or the levels of lipid droplets, LDAs, which are reservoirs of fatty acids for FAO, the invasiveness of cancer cells can be significantly impaired. This is because lipids are not only energy sources, but also components of cell membranes, signaling molecules and modulators of cell dynamics, like migration and invasion. When the supply or synthesis of lipids is disrupted, cancer cells lose the metabolic flexibility to support rapid growth, invasion, and adaptation to new microenvironments, which are hallmark features of cancer progression. These connections illustrate a complex biological network, where metabolism dictates not just energy availability, but also cellular behavior and genetic expression. The dependency of cancer cells on lipid metabolism for both energy and epigenetic regulation highlights potential vulnerabilities that can be targeted therapeutically. By modulating FAO or lipid synthesis pathways, or by altering the availability of acetyl-CoA for epigenetic modifications, there's an opportunity to disrupt the metabolic advantage cancer cells have, potentially slowing down tumor growth, metastasis, and perhaps even reversing some of the oncogenic epigenetic changes that drive cancer aggressiveness. Epigenetic modulation plays an integral role in the control of gene expression, particularly in the context of cancer, where metabolic shifts like those involving lipids can influence cellular behavior. One of the key approaches to target this modulation involves the use of BET, bromodomain and extraterminal inhibitors such as BET762. These inhibitors target bromodomain proteins, which are readers of acetylated histones. thereby playing a crucial role in chromatin remodeling and gene transcription. IBET-762 specifically disrupts the interaction between bromodomain proteins and acetylated lysine residues on histones, effectively altering the transcriptional landscape. In cancer cells, particularly those that have undergone lactate-induced lipid reprogramming, this inhibition can have profound effects. Lactate, often from cancer-associated fibroblasts, promotes lipid synthesis and storage in lipid droplets, LDS, which supports rapid proliferation and survival. By targeting the BET proteins BET-762 can interfere with this metabolic reprogramming, reducing the expression of genes that are pivotal for lipid metabolism and LD formation. This intervention leads to differential gene expression, particularly affecting genes associated with LDS like PLIN2, Paralipin2, which is crucial for ALT stabilization and lipid storage. The downregulation of such genes with BET inhibition suggests a mechanism through which cancer cells lose their ability to efficiently store lipids, thereby impacting their growth and adaptability. This shift in gene expression not only affects lipid metabolism, but can also influence broader cellular functions, including proliferation, apoptosis, and potentially the cancer cell's ability to metastasize. The connection between lipid metabolism and cancer metastasis is explored through both experimental and spontaneous metastasis models. In experimental setups, cancer cells exposed to lactate conditioning mimicking the influence of the tumor microenvironment show enhanced metastatic potential. This is partly due to the metabolic flexibility provided by lipid accumulation, which can fuel energy demands or provide materials for new membrane synthesis during the invasive process. However, when these cells are treated with BET inhibitors like IBET-762, there's a noticeable reduction in their metastatic capacity in viva. This decrease is linked to the metabolic changes induced by the inhibitor, where the cells are less capable of leveraging lipids for the energetic or structural support needed for metastasis. In spontaneous metastasis, models where tumors are allowed to grow and spread naturally, similar observations hold true. The inhibition of BET proteins reduces not just primary tumor growth, but also the incidence or extent of metastasis, suggesting that epigenetic control over lipid metabolism pathways can be a critical determinant of cancer's metastatic behavior. The implications of these findings are profound, illustrating how metabolic reprogramming and epigenetic regulation are intertwined in cancer progression. By targeting the epigenetic machinery that supports lipid metabolism, there's a potential not just for controlling tumor growth, but also for preventing or reducing metastasis, a major challenge in cancer treatment. This approach exemplifies how understanding the links between metabolism, epigenetics, and cancer biology can lead to innovative therapeutic strategies, aiming at the very foundations of cancer's aggressive behavior. Histological analysis, particularly through hematoxylin and eosin H&E staining, remains a cornerstone in the routine diagnosis of cancer, offering a visual representation of tissue architecture and cellular changes. H&E staining uses hematoxylin to stain nuclei blue, highlighting cellular proliferation and nuclear atypia, while eosin stains cytoplasm and extracellular matrix paint, revealing alterations in cellular morphology and tissue structure. This method is invaluable for identifying cancer-related changes like invasion necrosis or the presence of lipid droplets in cancer cells, which can be indicative of metabolic shifts associated with malignancy. The therapeutic implications of understanding cancer's metabolic landscape, particularly the mechanistic connections between lactate lipid accumulation and metastasis, are vast. Lactate produced in high amounts by cancer cells and cancer-associated fibroblasts not only acidifies the tumor microenvironment, but also fuels lipid synthesis, contributing to the formation of lipid droplets. These lipids are not just energy reservoirs, but also support membrane synthesis for cell division and migration, potentially aiding metastasis. By recognizing this, we can see how metabolic reprogramming underpins cancer's aggressive behavior, offering pathways for therapeutic intervention. This understanding leads to the development of pharmacological targets aimed at disrupting these metabolic pathways. For instance, targeting glutathione, the glucose transporter upregulated in many cancers to facilitate glucose uptake, could starve cancer cells of their primary energy source. Inhibiting hexokinase, the enzyme that phosphorylates glucose to trap it inside cells, would further disrupt glycolytic flux. Lactate dehydrogenase, LDH, which converts pyruvate to lactate, is another target. Its inhibition could reduce lactate production and the subsequent lipid synthesis. McTONA, monocarboxylate transporter 1, is crucial for lactate export and uptake, and by blocking this transporter, one could decrease the metabolic symbiosis between cancer cells and their stroma. Lastly, targeting hypoxia-inducible factor, HIF, which is activated under low oxygen conditions and drives many of these metabolic adaptations, could broadly impact cancer's ability to survive and proliferate under hypoxic stress. The conclusion drawn from these mechanistic insights highlights a strategy where cancer treatment can be tailored by targeting not just the cancer cells, but the metabolic ecosystem. They thrive in metabolic ecosystem. They thrive in all. Inhibiting these pathways could lead to a reduction in lipid accumulation, thereby limiting the energy and building blocks available for cancer growth and spread. This approach also has the potential to modulate the tumor microenvironment, making it less hospitable for cancer progression, possibly enhancing the effectiveness of other therapies like chemotherapy or immunotherapy by altering the metabolic landscape in which these treatments operate. By integrating histological insights with metabolic and genetic knowledge, we're moving towards a more nuanced understanding of cancer, where the visual cues from H&E staining guide us to the molecular and metabolic aberrations that can be therapeutically targeted. This holistic approach underscores the complexity of cancer, but also illuminates a path toward more effective personalized treatments that consider the unique metabolic adaptations of each cancer type or even each patient's tumor. Lecture 8. The physicochemical composition of the tumor microenvironment, TME, is a complex and dynamic ecosystem that significantly influences cancer cell behavior, treatment response, and disease progression. This environment is characterized by several key features, including acidity hypoxia, increased lactate levels, reduced glucose availability, and alterations in the secretome of both cancer and stromal cells. One of the predominant characteristics of the TME is its acidity. As cancer cells adopt a glycolytic metabolism, even in the presence of oxygen, a phenomenon known as the Warburg effect, they produce high amounts of lactic acid. This lactate is then exported into the extracellular space, reducing the pH and creating an acidic microenvironment. This acidity can inhibit immune cell function, degrade the extracellular matrix, and promote cancer cell invasion by activating proteases that break down barriers to metastasis. Hypoxia, or low oxygen levels, is another defining feature of the TME, particularly in the core of solid tumors, where the rapid proliferation of cancer cells often outpaces angiogenesis, the process of new blood vessel formation. Hypoxia leads to the stabilization of hypoxia-inducible factors, HIFs, which in turn trigger a cascade of responses, including further metabolic adaptation towards glycolysis, angiogenesis, and survival pathways that help cancer cells endure low oxygen conditions. This hypoxic environment not only promotes genetic instability, but also drives cells towards a more aggressive phenotype, enhancing their metastatic potential. The gradients of oxygen and nutrients within the TME are crucial for understanding how these physicochemical properties vary spatially. Oxygen and glucose concentrations decrease with increasing distance from the tumor's blood supply, creating a gradient where cells closer to blood vessels experience relatively better oxygenation and nutrient availability, while those further away face severe hypoxia and nutrient deprivation. Conversely, the concentration of hydrogen ions, HH, and lactate increases in these poorly perfused regions, contributing to the acidic environment that further challenges normal cell function, while favoring cancer cell survival and adaptation. These conditions also lead to changes in the secretome, the collection of proteins and other molecules secreted by cells into their environment. The time secretome is altered to include growth factors, cytokines, and extracellular matrix components that promote tumor growth, angiogenesis, and immune evasion. For instance, hypoxia can induce the secretion of VEGF, vascular endothelial growth factor, promoting new blood vessel formation, while acidic conditions might lead to the release of matrix metalloproteinases that facilitate invasion. The connections between these physicochemical characteristics are profound. They create selective pressures that can drive the evolution of cancer cells towards more aggressive phenotypes. The acidic and hypoxic conditions can select for cells with mutations or epigenetic changes that confer survival advantages under these stresses, leading to a more malignant tumor population. Moreover, the altered metabolic landscape influences not just the cancer cells, but also the behavior of immune cells, stromal cells, and the overall architecture of the tumor, making the time a critical target for therapeutic intervention. Understanding these gradients and characteristics opens up avenues for treatment strategies that aim to normalize the time, either by targeting the metabolic pathways that lead to its aberrant composition or by modulating the physical properties of the environment to enhance drug delivery immune response, or to specifically kill cancer cells adapted to these harsh conditions. This knowledge is pivotal in moving towards therapies that consider the tumor not as a collection of isolated cancer cells, but as an ecosystem where physicochemical properties play a significant role in disease progression and treatment efficacy. Oxygen, O1, is fundamental to life, serving as the final electron acceptor in oxidative phosphorylation, the process that generates the majority of ATP in eukaryotic cells. This reliance on oxygen for bioenergetics is not just a human trait, but is crucial across species from simple organisms to complex multicellular life forms. The availability of oxygen profoundly influences cellular metabolism growth and survival, making its sensing and regulation critical for adaptation to environmental changes. Hypoxia, or the condition of low oxygen availability, acts as a significant stressor in both physiological and pathophysiological contexts. Physiologically, hypoxia can be experienced at high altitudes or during physical exertion, prompting immediate responses like increased ventilation to boost oxygen intake. Pathophysiologically, hypoxia is a hallmark of many diseases, particularly in cancer, where it contributes to tumor progression, metastasis, and resistance to therapy due to the selective pressures it exerts on cells. To adapt to hypoxia, organisms and cells employ several mechanisms. One of the initial responses is the enhancement of ventilation to increase oxygen delivery to tissues. At the cellular level, hypoxia triggers angiogenesis, the growth of new blood vessels, from existing ones to improve oxygen supply. This is driven by hypoxia-inducible factors, HIFs, which are key players in the oxygen sensing pathway. HIFs, upon stabilization under low oxygen conditions, act as transcription factors that upregulate genes involved in angiogenesis, glycolysis, and survival under low oxygen, thus reprogramming metabolism towards anaerobic pathways to maintain ATP production when oxidative phosphorylation is compromised. The metabolic shift includes an upregulation of glycolytic enzymes and glucose transporters, preparing cells for increased glucose utilization in the absence of adequate oxygen. This adaptation is not without consequences. while it allows cells to survive and proliferate in low oxygen environments. It also contributes to the aggressive behavior of cancer cells, including invasion, metastasis, and resistance to treatment. The significance of these mechanisms was recognized with the 2019 Nobel Prize in Physiology or Medicine, awarded to William G. Kaelin, J.R. Sir Peter J. Ratcliffe, and Greg L. Semenza for their discoveries on how cells sense and adapt to oxygen availability. Their work elucidated the molecular details of how HIFs operate, revealing how oxygen levels regulate gene expression through the interaction of HIF with oxygen-dependent hydroxylases. Under normoxic conditions, these hydroxylases tag HIF for degradation. But in hypoxia, this degradation is inhibited, allowing HIF to accumulate and drive the transcription of genes that enable survival under low oxygen. The connections between oxygen-sensing cellular adaptation and disease are profound. Understanding these pathways not only sheds light on how organisms adapt to varying oxygen levels, but also provides insights into disease states where hypoxia plays a role, like cancer, ischemic heart disease, or stroke. This knowledge has spurred the development of therapies aimed at manipulating HIF pathways or targeting the metabolic consequences of hypoxia to treat or manage these conditions. Moreover, it underscores the intricate relationship between environment, metabolism, and genetics, illustrating how external conditions like oxygen availability can dictate internal cellular responses affecting health, disease, and evolution across life forms. Hypoxia, inducible factor. HIF is a pivotal protein complex in cellular oxygen sensing, orchestrating the adaptive response to low oxygen conditions or hypoxia. HIF primarily exists in three isoforms: HIF-1A, HIF-2A, and HIF-3A, each with unique roles in mediating the hypoxia response across different tissues and during various physiological and pathological conditions. The structure of HIF involves an alpha, A, and a beta, delta, subunit. Under normoxic conditions, the A subunits, HIF-1A, HIF-2A, are rapidly degraded due to hydroxylation by prolyl hydroxylases, PHDs, also known as egonon-3. This hydroxylation targets HIF-A for ubiquitination by the von Hippel-Lindau VH tumor suppressor protein, leading to its proteasomal degradation. However, when oxygen levels drop, this degradation process is inhibited, allowing HIF-A to accumulate. It then translocates to the nucleus, where it heterodimerizes with the stable beta subunit, known as ARNT, aryl hydrocarbon receptor nuclear translocator, forming an active transcription factor. This HIF-ECHA ARNT dimer recognizes and binds to hypoxia response elements, EVs in the DNA, leading to the transcriptional activation of hundreds of genes. These genes are involved in angiogenesis, e.g., glucose, metabolism, e.g., Rutona glycolytic enzymes, erythropoiesis, EPO, pH, regulation and cell survival mechanisms, thereby facilitating adaptation to low oxygen environments. HIF-1A tends to be more ubiquitously expressed and involved in acute responses to hypoxia, while HIF-2A plays a more prominent role in chronic hypoxia, influencing processes like angiogenesis in specific tissues. HIF-3A's role is less understood, but can act as a negative regulator of HIF activity. The pathway involves several key proteins beyond the HIF subunit SU. The PAGs, egonon-3, are oxygen sensors that hydroxylate HIF-A on specific proline residues, marking it for VAL-mediated degradation. Factor inhibiting HIF, Phe1, hydroxylates an asparagine residue on HIF-A, blocking its interaction with co-activators like P300 and CBP, which are essential for transcriptional activation. P300 and CBP not only co-activate HIF, but also modify chromatin structure to facilitate gene transcription. The complexity of the HIF pathway underscores its central role in cellular adaptation to hypoxia. In cancer, for example, HIF activation leads to a metabolic shift towards glycolysis, promoting survival in the hypoxic tumor microenvironment and enhancing tumor aggressiveness by promoting angiogenesis and metastasis. In other contexts like high-altitude adaptation or ischemic responses, HIF's regulation of genes like EPO can lead to increased red blood cell production or angiogenesis to restore oxygen delivery. The connections here are multifaceted. HIF not only responds to oxygen levels, but also modulates them through its effects on metabolism and angiogenesis. This feedback loop between oxygen-sensing gene expression and cellular behavior has profound implications for health and disease. Understanding these pathways has opened up avenues for therapeutic intervention, where modulating HIF activity could treat conditions characterized by excessive or insufficient angiogenesis, metabolic disorders, or to enhance tissue repair in ischemic conditions. The Nobel Prize recognition in 2019 for the elucidation of these mechanisms highlights their foundational importance in biology and medicine, illustrating how fundamental discoveries can lead to significant advancements in human health. The regulation of HIF-1 stability is central to the cellular response to hypoxia, orchestrating a wide array of adaptive processes. Under normoxic conditions, HIF-1 stability is tightly regulated by prolyl hydroxylase, domain-containing proteins, PHDs, specifically P8-DONE, PHD2, and PHT3. These enzymes use molecular oxygen to hydroxylate specific proline residues in HIF-1 a reaction that requires 2-oxoglutarate, also known as ketoglutarate, iron, Fe, and ascorbate, vitamin C, as cofactors. Once hydroxylated, HIF-1 is recognized by the von Hippel-Lindau VAL tumor suppressor protein, which acts as an E3 ubiquitin ligase, targeting HIF-1 for proteasomal degradation, thus maintaining low levels of HIF-1 in the presence of oxygen. However, under hypoxic conditions, the limited availability of oxygen reduces PHD activity, preventing the hydroxylation of HIF-1. As a result, HIF-1 is not ubiquitinated by EHL and escapes degradation, leading to its accumulation in the cytosol. Stabilized HIF-1 can then translocate to the nucleus, where it dimerizes with ARNT, HIF-1 to form the active transcription factor. This complex binds to hypoxia response elements in DNA, initiating the transcription of genes that promote survival in low oxygen environments, including those involved in angiogenesis, glycolysis, and erythropoiesis. The connection between PHDs and metabolic regulation adds another layer to HIF-1's regulation. PHD enzymes are not only sensitive to oxygen, but also to the availability of their cofactors, particularly 2-oxoglutarate, which is an intermediate in the TCA cycle. The concentration of 2-oxoglutarate can influence PHD activity, thereby linking cellular metabolism directly to HIF regulation. For instance, in cancer cells where the TCA cycle might be reprogrammed, the levels of 2-oxoglutarate could be altered, affecting PHD's ability to hydroxylate HIF-1. This creates a feedback loop where metabolic changes can influence oxygen sensing when the TCA cycle is functioning normally. It produces 2-oxoglutarate promoting PHD activity and HIF-1 degradation. However, if the cycle is disrupted, or if there's an increase in succinate or fumarate, other TCA intermediates that can inhibit PHDs, HIF-1 stabilization might occur even in normoxia, mimicking a hypoxic response. In conditions where glucose or glutamine metabolism is high, leading to an accumulation of TCA cycle intermediates, PHD activity can be modulated. providing a mechanism where metabolism can dictate oxygen-sensing responses. This metabolic link to HIF regulation highlights how cellular energy states, or nutrient availability, can affect oxygen sensing, providing a mechanism for cells to adapt, not just to low oxygen, but also to metabolic stress. It also underscores the complexity of cancer biology, where tumors can exploit these pathways for survival and growth even in environments where oxygen supply is adequate. Understanding these connections offers potential therapeutic targets where modulating PHD activity or the availability of its cofactors could influence HIF-driven gene expression, potentially controlling tumor metabolism, angiogenesis, or even promoting tissue repair in ischemic conditions. This intricate interplay between oxygen-sensing metabolism and gene regulation exemplifies the adaptive nature of cellular responses to environmental and internal cues. Hypoxia, inducible factor. HIF orchestrates a broad spectrum of adaptive responses by regulating the expression of numerous target genes pivotal for survival under low oxygen conditions. Among these, erythropoietin, EPO, is crucial for erythropoiesis, stimulating the production of red blood cells to enhance oxygen carrying capacity. Vascular endothelial growth factor, VEGF, promotes angiogenesis, the formation of new blood vessels to improve oxygen and nutrient supply to hypoxic tissues. Pyruvate dehydrogenase kinase 1, PKONE, plays a role in metabolic reprogramming by inhibiting the pyruvate dehydrogenase complex, thereby favoring glycolysis over oxidative phosphorylation in the absence of adequate oxygen. In the context of cancer, HIF's role extends beyond simple adaptation to hypoxia, influencing several aspects of tumor biology that contribute to tumorigenesis. One significant impact is in cancer stem cell specification. HIF can promote the maintenance or generation of cancer stem cells, which are implicated in tumor initiation, metastasis, and resistance to therapy due to their self-renewal capabilities, and pluripotency. This is mediated through the upregulation of genes that support stemness, survival, and proliferation under stress. HIF also plays a critical role in immune evasion. By activating genes that modulate the immune microenvironment, such as those encoding for immune checkpoint inhibitors or immunosuppressive cytokines, tumors can escape immune surveillance. This creates a permissive environment for tumor growth and metastasis, where the immune system's ability to recognize and eliminate cancer cells is compromised. The metabolic adaptation driven by HIF in cancer cells is profound. HIF upregulates numerous glycolytic enzymes like glucose transporters, e.g., glutathione, hexokinase, and phosphofructokinase, shifting metabolism towards glycolysis even in the presence of oxygen, a phenomenon known as the Warburg effect. This adaptation not only provides cancer cells with a means to generate ATP under hypoxic conditions, but also supplies intermediates for biosynthetic pathways essential for rapid cell division. Additionally, HIF modulates oxidative phosphorylation by regulating genes like PKONE, which reduces the conversion of pyruvate to acetyl-CoA, favoring the accumulation of glycolytic intermediates. These metabolic changes are not just about energy. They support the aggressive behavior of cancer cells. By altering metabolism, HIF influences the tumor microenvironment, creating conditions that favor tumor growth, invasion, and metastasis. The acidic environment resulting from lactate production can degrade the extracellular matrix, facilitating cancer cell motility, while the metabolic stress can select for cells with increased survival capabilities under nutrient and oxygen deprivation, further driving tumor evolution. The connections between HIF, its target genes, and cancer biology illustrate a complex network where environmental cues like hypoxia are translated into genetic and metabolic responses that support cancer progression. Understanding these pathways not only deepens our insight into how tumors adapt and survive in hostile environments, but also offers targets for therapeutic intervention. By targeting HIF or its downstream effects, therapies could potentially disrupt the metabolic and immune advantages tumors gain, reducing tumor aggressiveness, enhancing immune responses against cancer, or sensitizing cancer cells to existing treatments. This knowledge underscores the importance of considering the tumor microenvironment in cancer therapy, aiming for strategies that not only target cancer cells, but also the supportive ecosystem they create. Tumor hypoxia, a condition where oxygen levels are insufficient within the tumor mass, arises as cancer grows beyond the capacity of its initial blood supply to provide adequate oxygen diffusion. This oxygen deprivation activates hypoxia inducible factor HIF, particularly HIF-1A and HIF-2A, initiating a cascade of cellular responses aimed at survival and adaptation. One of the primary adaptive mechanisms is angiogenesis, the formation of new blood vessels from pre-existing ones, which is orchestrated by HIF through the upregulation of genes like vascular endothelial growth factor, VEGF. As tumors expand, areas of hypoxia become more pronounced, not only due to the distance from existing blood vessels, but also because of the chaotic and often inefficient vascular network that develops within tumors. This hypoxic adaptation is not merely about survival. It prepares cancer cells for more aggressive behaviors like invasion and metastasis. Hypoxia-selected cancer cells can exhibit increased motility, invasiveness, and resistance to apoptosis traits that are advantageous for disseminating to distant sites. Moreover, the hypoxic environment can lead to genetic instability, promoting further mutations that might enhance tumor aggressiveness. Given its pivotal role in cancer progression, HIF has emerged as a significant therapeutic target. However, targeting HIF directly has proven challenging. HIF inhibitors have faced significant hurdles, including issues with toxicity and specificity, leading to several clinical trial failures. These inhibitors often affect not just cancer cells, but also normal cells that rely on HIF for homeostatic functions, like response to physiological hypoxia or tissue repair, resulting in side effects that limit their therapeutic window. Despite these challenges, a success story in this domain is belzutifan, a selective inhibitor of HIF-2A, which received FDA approval for treating von Hippel-Lindau (VL) associated renal cell carcinoma, RCC. VL disease involves mutations in the VL gene, leading to constitutive HIF-2A activity, which drives tumor formation. Belzutifan's approval marks a significant milestone, demonstrating that targeting specific isoforms of HIF can be effective in certain genetic backgrounds where HIF is aberrantly activated. Looking to the future, the development of HIF inhibitors is expected to focus on combination therapies to overcome resistance and enhance efficacy. Understanding the mechanisms through which cancer cells adapt to or circumvent HIF inhibition is crucial. This includes exploring how tumors might switch between HIF-1A and HIF-2A, or how they might compensate through other pathways when one is blocked. There's also interest in developing more specific HIF-1A inhibitors, as this isoform is more universally involved in the hypoxic response across different cancer types. The connections here are intricate. Targeting HIF isn't just about blocking one pathway, but understanding and manipulating a network where oxygen sensing, metabolism, angiogenesis, and immune evasion intersect. The success of therapies like belzutifan opens the door to personalized medicine, approaches based on the genetic or metabolic profile of a tumor. Moreover, this work underscores the complexity of tumor biology, where environmental pressures like hypoxia drive not just survival, but the evolution of cancer towards more aggressive and resistant phenotypes. As research progresses, the aim is to tailor treatments that can disrupt these adaptive mechanisms, potentially leading to more effective cancer management strategies. Like the concept of metabolic reprogramming in cancer, particularly the Warburg effect, has evolved significantly since Otto Warburg first described it nearly a century ago. Warburg cancer cells predominantly convert glucose to lactate even in the presence of oxygen, a phenomenon now known as aerobic glycolysis. This shift was initially interpreted as a defective mitochondrial function, but contemporary research has painted a more nuanced picture of tumor metabolism. The historical context of the Warburg effect reminds us that what was once seen as a fundamental flaw in cancer cell metabolism might actually represent a strategic adaptation. Warburg's observations set the stage for understanding how cancer cells could sustain rapid proliferation by favoring glycolysis, which not only generates ATP quickly, but also produces biosynthetic intermediates necessary for cell growth and division. However, this understanding has been revisited with modern insights, revealing that the metabolic landscape of tumors is far more complex than simple glycolysis. Current understanding acknowledges that while many tumors do exhibit the Warburg effect, this does not mean all cancer cells, or even all parts of a tumor, uniformly adopt this metabolic strategy. Tumor tissues often display a heterogeneous metabolic profile, where some cells or regions might engage in significant or even elevated levels of glucose oxidation. This suggests that mitochondrial respiration in cancer cells is not always impaired but can be selectively used depending on the tumor's environment, nutritional status, or stage of progression. This variability is partly due to the existence of different subpopulations within tumors, each potentially adopting distinct metabolic programs. Some cancer cells might rely heavily on glycolysis for rapid ATP production and to accumulate biosynthetic intermediates, while others, particularly those in less hypoxic or nutrient-rich regions, might utilize oxidative phosphorylation to maximize energy yield from glucose. This heterogeneity can be driven by genetic mutations, epigenetic modifications, or microenvironmental cues like oxygen and nutrient availability, leading to a dynamic metabolic landscape where cells can switch strategies in response to stress or therapeutic pressures. The revisiting of the Warburg effect with these modern insights has implications for cancer therapy and research. Recognizing that not all cancer cells are glycolytic opens up the possibility of targeting metabolic pathways other than glycolysis, such as mitochondrial function or lipid metabolism. It also highlights the potential for therapeutic resistance, where tumor cells might adapt their metabolism in response to treatments targeting one pathway, switching to another for survival. The connections between these insights and cancer biology are profound. Metabolic reprogramming is not just about energy. It's about survival, proliferation, and adaptation in a hostile environment. This understanding has led to the exploration of combination therapies, where targeting multiple metabolic pathways could prevent cancer cells from compensating through alternative routes. Moreover, it underscores the importance of personalized medicine approaches, where the metabolic profile of a patient's tumor could guide treatment decisions, aiming not just at killing cancer cells, but at disrupting the metabolic ecosystem that supports tumor growth and metastasis. In essence, revisiting the Warburg effect a century later has transformed our approach from viewing it as a singular metabolic anomaly to recognizing it as part of a broader, more flexible metabolic adaptation strategy in cancer, thereby expanding the avenues for therapeutic intervention. Metabolic heterogeneity in cancer is a testament to the adaptability and complexity of tumor biology, where no single metabolic strategy can fully describe the energy and biosynthetic needs of cancer cells. Evidence from biopsies has shown that there's a selective pressure against mutations that impair the TCA cycle in cancers, indicating that while the Warburg effect describes a glycolytic predominance, the TCA cycle remains crucial for producing intermediates essential for anabolism. These intermediates are building blocks for nucleotides, amino acids, and lipids, which are vital for the rapid proliferation of cancer cells. Thus, despite the observed glycolytic shift, the TCA cycle often maintains a significant role, suggesting that cancer metabolism is more about balancing multiple pathways rather than relying solely on one. The concept of metabolic flux in cancer cells underscores this balance. Cancer cells do not just favor glycolysis over oxidative phosphorylation, OXPHOS. They optimize both pathways for their unique demands. The TCA cycle is not only about energy production, but also about generating biosynthetic precursors. Even under conditions where glycolysis is upregulated, some cancer cells continue or even enhance TCA cycle activity to support anabolism, showing that the relationship between glycolysis and OXPHOS is not a simple switch, but a coordinated effort where both pathways are enhanced to meet the tumor's growth requirements. There's a common misconception that cancer cells abandon oxidative phosphorylation in favor of glycolysis. However, this is not universally true. Many tumors exhibit an increase in both glycolysis and OXPHOS, often referred to as a hybrid or dual metabolic phenotype. This dual engagement allows cancer cells to maximize ATP production, handle oxidative stress, and ensure a supply of biosynthetic intermediates. In some cases, cancer cells might even upregulate mitochondrial biogenesis to support higher levels of OXPHOS, particularly in oxygen-rich areas of the tumor or when nutrients are plentiful. The connections between these metabolic strategies reveal a sophisticated level of cellular adaptation. Cancer cells can adjust their metabolism in response to microenvironmental cues like oxygen availability, nutrient supply, or therapeutic interventions. For instance, in hypoxic regions they might lean more towards glycolysis, while in normoxic areas or when reoxygenated post-treatment, they might ramp up OXPHOS. This metabolic flexibility not only supports tumor growth and survival, but also contributes to resistance against treatments that target one metabolic pathway. Understanding this metabolic heterogeneity has profound implications for cancer therapy. It suggests that effective treatments might need to target not just one pathway, but the metabolic adaptability of cancer cells. This could involve combinations of drugs that inhibit glycolysis, OXPHOS, or other metabolic pathways, or strategies that exploit the metabolic differences between cancer and normal cells, like selectively targeting the enhanced OXPHOS in certain cancer cells, or the dependency on glycolysis in others for energy. This nuanced view of cancer metabolism challenges the traditional narrative of the Warburg effect, highlighting instead a dynamic metabolic landscape where cancer cells leverage both ancient and modern metabolic pathways to thrive, grow, and resist therapies. Recognizing and targeting this metabolic diversity could lead to more personalized and effective cancer treatments, tailored to the metabolic profile of each patient's tumor. Metabolic flexibility is a hallmark of cancer, reflecting the ability of cancer cells to adapt their metabolism to utilize a broad spectrum of nutrients beyond just glucose, including glutamine lipids and various amino acids. This adaptability allows cancer cells to thrive under diverse nutritional conditions and microenvironmental stresses, showcasing a spectrum from glycolytic preference to reliance on oxidative metabolism. Cancer cells exhibit heterogeneous metabolic preferences, where some subpopulations within a tumor might favor one nutrient source over another, based on availability or genetic alterations. For instance, glutamine can become a primary carbon and nitrogen source when glucose is limited, supporting both energy production through the TCA cycle and biosynthesis. Lipids, particularly through fatty acid oxidation or lipid droplet storage and utilization, provide an alternative energy source and building blocks for membrane synthesis in cancer cells. Similarly, other amino acids are not just building blocks for proteins, but are also involved in one-carbon metabolism redox balance and signaling pathways that support proliferation and survival. This Metabolic flexibility is underpinned by symbiotic and parasitic relationships within the tumor microenvironment. Cancer cells can engage in nutrient scavenging or metabolic symbiosis with stromal cells like fibroblasts or adipocytes. For example, cancer-associated fibroblasts might produce lactate or other metabolic byproducts that cancer cells can recycle, while cancer cells might secrete factors that induce nutrient release from surrounding cells. This interaction creates a dynamic nutrient landscape where cancer cells can exploit the metabolic products of their neighbors, enhancing their survival and growth in nutrient-poor environments. Contrary to the traditional view of cancer metabolism, focusing solely on glycolysis, mitochondria play a pivotal role in this metabolic reprogramming. They are not just the sites for oxidative phosphorylation, but are also central to the metabolism of glutamine fatty acids and amino acids. Mitochondria can adapt to support both high glycolytic and oxidative metabolic states, contributing to the production of ATP, intermediates for biosynthesis and regulation of cellular redox status, which is crucial for cancer cell survival under oxidative stress. The demand for amino acids in cancer goes beyond protein synthesis. Amino acids, like glutamine, serine, and glycine, are essential for energy production, nucleotide synthesis, and maintaining redox homeostasis. Glutamine in particular stands out for its versatile role in cancer metabolism. It can be converted to glutamate, which then enters the TCA cycle, supporting both ATP production and the synthesis of nucleotides and other amino acids. Glutamine metabolism is also linked to cancer's ability to manage oxidative stress through the provision of glutathione precursors, and to support rapid proliferation by fueling the biosynthesis of nucleic acids and lipids. The connections between these metabolic adaptations are profound. They illustrate how cancer cells can switch or balance between different metabolic pathways to optimize survival and growth under varying conditions. Understanding this metabolic landscape opens avenues for therapeutic strategies that target not just one aspect of cancer metabolism, but aim to disrupt the cancer cell's ability to adapt and thrive. This could involve targeting glutamine metabolism, lipid synthesis, or oxidation, or the interactions with the tumor microenvironment that facilitate nutrient acquisition and metabolic symbiosis. Such strategies might leverage the metabolic vulnerabilities unique to cancer cells, potentially leading to more effective, personalized cancer treatments. Glutamine metabolism in cancer has been a subject of intense research for over half a century, revealing its critical role in the metabolic landscape of tumors. Cancer cells exhibit a profound dependency on glutamine, often consuming it at rates far exceeding those of normal cells, a phenomenon first noted in the 1950s by Harry Eagle. This high consumption underscores glutamine's multifaceted role in cancer biology, serving not just as a nutrient, but as a central node in the metabolic network, supporting cancer cell proliferation, survival, and adaptation. Glutamine serves multiple functions in cancer. It's pivotal for bioenergetics, acting as an alternative energy source through its conversion to glutamate and subsequent entry into the TCA cycle. This process, known as glutaminolysis, where glutamine is deaminated to glutamate and then converted to ketoglutarate, provides carbon skeletons for both ATP production and the synthesis of other biomolecules. Glutamine also plays a significant role in biosynthesis, providing nitrogen for the synthesis of nucleotides, amino acids, and non-essential amino acids, thus fueling rapid cell division. As a nitrogen storage and transport molecule, glutamine can shuttle nitrogen between organs or within cells, supporting various synthetic pathways. The pathways of glutamine metabolism in cancer are diverse and interconnected. Glutaminolysis leads to the production of glutamate, which can be further metabolized to ketoglutarate, feeding into the TCA cycle. This pathway supports energy production, but also serves as an anaplerotic reaction, replenishing TCA cycle intermediates that are siphoned off for biosynthesis. Glutamine is also crucial for glutathione synthesis, an antioxidant, critical for managing the oxidative stress that cancer cells frequently encounter due to their rapid metabolism. The conversion of glutamine to glutamate also generates ammonia, which can have both local and systemic effects, contributing to acidification of the tumor microenvironment or altering systemic pH balance. Glutamine acts as a metabolic hub, influencing both carbon and nitrogen flux within the cell. In the cytosol, glutamine's nitrogen can be used for nucleotide and amino acid synthesis, while in mitochondria it contributes to energy production via the TCA cycle. This dual functionality highlights glutamine's unique position in cancer metabolism, capable of supporting both oxidative and reductive pathways. Anaplerosis, or the replenishment of TCA cycle intermediates, is directly supported by glutamine, ensuring that the cycle can continue even when intermediates are used for biosynthesis. Conversely, reductive carboxylation, where aminoacetoglutarate is converted back to citrate outside the TCA cycle, uses glutamine-derived carbon for lipid synthesis, particularly under hypoxic conditions or when oxidative phosphorylation is less favorable. The connections between glutamine metabolism and cancer biology are profound, illustrating how this amino acid supports the metabolic flexibility required for cancer's aggressive growth. By targeting glutamine metabolism, there's potential to disrupt multiple aspects of cancer cell biology, from energy production to antioxidant defense, from DNA synthesis to lipid membrane formation for new cells. This understanding has led to the exploration of glutamine antagonists, or inhibitors of glutamine-utilizing enzymes, as potential cancer therapies, aiming to starve cancer cells of this vital nutrient or interfere with its metabolic contributions. Such strategies reflect the ongoing quest to exploit metabolic vulnerabilities in cancer, leveraging our deep dive into the metabolic intricacies that support cancer's relentless growth and survival. The dependency on glutamine in cancer is not uniform, but rather exhibits significant heterogeneity across different types of cancer stages of tumor progression, and even within different cells of the same tumor. This variability reflects the diverse metabolic strategies cancer cells can employ to meet their needs for growth, survival, and adaptation. Some cancers, like certain subtypes of lung or pancreatic cancer, are notably glutamine addicted, relying heavily on glutaminolysis for energy and biosynthesis, whereas others might show less dependence, indicating that glutamine metabolism's role can vary greatly. The context in which cancer cells are studied also influences their glutamine requirements. In culture, conditions where nutrients are typically abundant, many cancer cell lines are seen to consume glutamine at high rates for both energy and biosynthetic purposes. However, in vivo, the tumor microenvironment presents a more complex scenario, with varying nutrient availability, oxygen levels, and interactions with stromal cells, which can modulate glutamine dependency. For instance, in hypoxic regions of tumors where oxygen is scarce, cancer cells might shift their metabolism towards glycolysis, potentially reducing their reliance on glutamine for energy, but still using it for biosynthesis or redox balance. This environmental adaptation underscores the plasticity of cancer metabolism, where the necessity for glutamine can be context-dependent. This heterogeneity in glutamine needs across cancers and conditions highlights the importance of an integrated view of cancer metabolism. Cancer cells do not rely solely on one metabolic pathway, but rather operate on a platform where glycolysis, oxidative phosphorylation, and amino acid metabolism, including glutamine, are all leveraged to support proliferation. Glycolysis provides quick ATP and biosynthetic intermediates. Oxidative phosphorylation offers efficient ATP production when oxygen is available. And amino acid metabolism, particularly glutamine, supplies nitrogen and carbon for macromolecular synthesis and energy under various conditions. The conclusion drawn from these insights is that to effectively target cancer metabolism, one must consider this integrated metabolic network. This network allows cancer cells to adapt to nutrient or oxygen limitations, therapeutic pressures, or changes in the microenvironment, making a one-size-fits-all approach to metabolic therapy challenging. However, it also opens up multiple fronts for therapeutic intervention. Targeting glutamine metabolism could be effective in glutamine-addicted cancers, but in others, combinations of therapies that also address glycolysis, mitochondrial function, or even the uptake and utilization of other amino acids might be necessary. Understanding this metabolic platform for cancer growth not only informs the design of new cancer therapies, but also underscores the importance of personalized medicine in oncology. By mapping the metabolic profile of a patient's cancer treatments, can be tailored to disrupt the specific metabolic pathways that the cancer relies on for growth, potentially leading to more effective outcomes. This holistic approach to cancer metabolism, acknowledging its complexity and adaptability, is crucial for advancing cancer treatment strategies beyond traditional methods, aiming towards therapies that can adapt as dynamically as the cancers they aim to control.